showing up.
Yeah, that's how it goes.
I don't, nobody else
I guess one of us also needs to unmute. Probably you. I think you.
Okay.
You
Is
should.
this good?
Yeah. Yeah. Are we getting feedback?
Is there still feedback? Yes.
It's just I need to have the sound up or something. Can you try now?
Yeah, good.
If you just move the screen up, fine.
Just hold it. Okay.
Okay. I think we're all set up. So yeah, first of all, thank you so much to the organizers for inviting me. It's great to be here. And thank you to all of you who are here, especially from the West Coast if it's 6:30 in the morning for you. I really appreciate it. And thanks to the organizers for giving me a little excuse for some of my axes labels being potentially too small. Now I can claim it's not my fault. Okay, so I always kind of start with the very high level goals of the work in my lab, which has focused for the past 10 years on understanding how differences in our personal genome sequence lead to individual differences in phenotype, both molecular phenotype and disease level phenotype, using various forms of machine learning, ranging from probabilistic modeling to now deep neural nets and all sorts of other. What's not on?
It's not working in the back either, in the poster area either. They said it was working earlier.
Yeah, we did.
What would you like me to?
Someone's coming in right now, I think, from the static, right now.
Yeah, let me find it again.
Or gesturing.
Yeah.
I did once give a talk at ASHG where none of the figures were showing up on one of the projectors, and so I had to just describe all of those results in detail. I could do that. Yeah, like imagine it's up and to the right.
Sorry,
You wanna try it on my laptop?
Sure.
That was working. I want to say you get what you pay for, but
So yeah, I don't even see it like on the.
and you're definitely sharing fine because I'm getting it.
You were getting it?
Here.
Okay, so do you want to try?
Oh, it's probably that this connection is not working, I guess, because that's going through Zoom. So,
Should I like?
yeah, just try plugging that
So
in.
this is powering the ones back there. Is that the deal?
It says choose to mirror instead from the little
Like, is that showing my desktop?
Yeah. I wonder if that's something in here where we can choose some option. No. Okay, let's just see what happens if we plug it into mine, I guess.
Okay.
Is that showing you're already muted, good. I think that's.
Okay, are we good? Restart?
Oh,
think-
they've got presented.
There you go, presenter mode.
Okay. If maybe does layout do it? Oh no, that's because this is the Zoom controls. We want the
If we just start it like this, it might actually
they still have presenter mode though.
presenter mode.
All listed. Okay, that I think it's not because you were you doing keynote?
No, so
Okay.
I did presenting mode first and then shared the screen.
Okay,
So
you
you
want
try that first. So stop,
Yeah,
stop
you
down
want it.
and Zoom. So. Sorry. Now if I go to your PowerPoint. Okay. You want presenter mode, yeah?
No, I don't need anything.
Oh, really?
Like if it just shows the.
Anything
regular
above.
mode, that's fine.
Okay.
The point is we don't want them to see presenter mode.
Yeah, oh, okay, I hear you.
Oh, I need to mirror if we need to mirror displays, that's what we'll do this. It's doing extended displays. So if we mirror displays instead, then it won't have the it won't have the
Presenter
option.
mode still? So you still have to connect, you can't just do it through Zoom for those ones?
Apparently.
Apparently not.
So that's not going through Zoom back there? Is that-
No, this goes through the
It's just that they don't, oh, okay, that's the problem, okay.
Oh, is there a
Still.
how is it still? I don't understand how it's doing it. But if it's set this to
Okay. And now.
mirror. And display and mirror.
Now I use the presenter mode here, yeah. That's still a bit of it.
How does it's forcing it into. Is there a way we can switch which one is presenter? Oh, we'd have to point on that one.
Yeah,
It's true.
okay, I can do that. Do you want me to send you a?
That seems like a painful way to have to do it. But
I mean, that might be quick from your PowerPoint.
I can't, and now I don't have a pointer.
Well, I can't point at anything. Mouse on the screen.
I did just send a PDF to the same
Okay.
box if you want to try.
Yeah. That how much
I'm
of
not
that?
going to show that screen again, so we're good.
Yeah.
Probably.
I think my problem now is when I connect this, it's making that the main display. So,
It's making.
okay, okay. Or it's trying to use that as the main display now. I need to be able to look at that to be able to fix this. But I can't plug in and walk over. Okay. Because now the setting I got it on with trying to get it to do that. I was trying to make that be the main display because I thought that would fix it. Yeah, I know. Peter, can you tell me where I'm trying to get my mouse to point at something so I can get that fixed. Okay. Is it showing the settings? Okay. So can you tell me where to move the mouse so I can?
stop. Right, right now. A little bit more. Up. Click.
Okay.
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 Wait,
Right.
wait.
Well, I want to set it right now. This isn't showing anything on this screen, so I just want to make that screen be the extended. That's the Creston. I want that to be the extended or mirrored. I need my laptop to be the main screen again. I was trying to. Do you know how to do that? That's function. How to get it to show all the screens? No.
Yeah, story.
Maybe unplug it and minimize all your other things and then.
Yeah, no. When I plug it back in, I still don't. Everything goes back to that screen.
Like he can't see anything up here. Yeah.
Yeah. Yeah.
This is very nice. What would you like to do?
So what's the issue if we go off yours? We don't get.
We don't get those screens at all, which is like, okay, and maybe not the end of the world.
Yeah. So if. Yeah. Yeah.
I can show mine from PDF and like.
Yeah, but it won't show on there, right? Because yours wasn't
I can
connecting.
plug it into it.
Yeah. Okay. Well, we'll try using Alexis's laptop again. See if that. Okay.
It's some light supposed to come on.
Yeah, that might come on, which was.
Are you seeing anything over there now?
Nothing. Too narrow and.
Yeah, actually, maybe you need to go to display settings.
Got it, no.
Nice, okay.
Yeah.
Okay, but
Okay.
I'm probably not on. Dude.
Oh, yeah, because you're not, right, you need to be sharing. Okay. Yeah, I know.
Can everyone actually see something now? Yes? Thank you so much for bearing with me. I appreciate it. All right, so as I was saying, I always start with the high-level goals of the lab, which is to understand how individual differences in our genomes propagate to individual differences in our cellular phenotypes, all the way up through disease risk and other high-level traits, using a whole variety of different machine learning and statistical methods. We all know that we're living in a time of genomic data explosion, but I gave a talk recently at Hopkins kind of looking back on my career, and this plot came up when I was kind of looking around, showing what I want to emphasize today, which is the scale of essentially population scale studies, right? And when I started my faculty career, I'm kind of going to approximate, the biggest GWAS were on the order of like 1,000, which is like basically zero when you're talking about association studies or power to build predictive models. And it has truly, exploded during my time as faculty. So all those things that I wrote in my research statement when I was applying for the job at Hopkins have like actually become possible during my time there. And I thought this really summarized that well. So today I'm going to talk about what that means for understanding the personal genome and personal functional omics in terms of, where are we really going with this? Are we anywhere close to being able to really deploy individualized medicine based on genomics? So what biological insights, risk predictors, precision treatments are really possible using omic data along with clinical information? So I'm kind of going to review where we are now in a couple of problems, but think more about what do we need to do from here? What are the data needs that we have, and what are the modeling strategies that might allow us to achieve the full potential? So some key questions that I'd like you to keep in mind are, do we really need what we're now calling AI, which I'm going to use to mean large nonlinear machine learning models? Do we really need that in personal genomics? Do we have the right data if we do need that? And if so, what are the architectures and training strategies that might be specific to biology and genetics that we could be considering? And I really am gonna present two different projects and think about the different properties of these two different data types, right? But one, if we're starting with DNA, where we are faced with problems such as small effect sizes, a long tail of effects from common variants, in addition to lots and lots of rare variants. DNA, of course, being largely constant over the lifespan, and confounders are somewhat constrained to things like linkage disequilibrium and population stratification. And I think that other data types and other problems have different considerations. So functional omics, we generally have larger effect sizes, so we can detect differential expression at much lower sample sizes, for example. But it's variable over time. The confounders are, in my experience, much more extensive and hard to interpret, reverse causality, biological and technical variation, all sorts of things. In addition to, especially for single cell and other data types, we're facing issues of sparsity and noise. But we can also take into account, perhaps, prior knowledge. So thinking about these two, in my opinion, quite different settings and how they lend themselves to machine learning methods, I'm going to talk about two different projects entirely today. So the first is using the personal genome, so DNA sequence, in prediction of gene expression. So why might we want to do this? I've worked for a long time in the interpretation of rare genetic variants, and here some of the primary applications included diagnosis of rare disease, where currently something like 50% of individuals go undiagnosed. Also thinking about using a prediction of gene expression as a building block towards using whole genome sequencing in trait prediction beyond just common variants. And finally, these models can presumably help us to understand the regulatory grammar in the human genome. But this problem is quite hard, and one of the figures that I've been showing for too many years is simply the number of rare variants that we have in each of our genomes. So healthy individuals are generally walking around with something like 50,000 or more variants below a minor allele frequency of 0.01. So if we really want to take all those into account, we have a lot of work to do. So linear models and association testing, univariate models even, can capture the effects for common previously observed variants and genes in measured context, right? So we can run EQTL studies like we did in GTEx. We could run TWAS and other types of analyses to identify what are the common variants and their effect sizes, even how they work together maybe in simple models in terms of how they affect expression of a gene in a measured context. Of course, these models don't consider the DNA sequence directly or the biological context of the variant, and in general, don't generalize beyond the variants that they saw in the original training set, or to rare variants, or to contexts or phenotypes where the association study was not run. So a big question on many people's mind, including in this room, is can we use large nonlinear machine learning methods to predict functional and phenotypic impact of new variants and really full individual DNA sequence? So sequence to function models are under a very active exploration now and hold a lot of promise to take DNA sequence as input and help us predict gene expression to understand the impact of rare genetic variation, not only common variation, in addition to helping us characterize disease regulatory mechanisms and potentially to identify causal variants, both for molecular phenotypes and even disease. So lots of models have been coming out in recent years that take DNA sequence as input and often in a multitask framework use deep neural networks based on convolution and transformers and other architectural choices to predict gene expression, DNA accessibility, histone modification, and other regulatory phenotypes that we have available from large data sets such as ENCODE or others, generally looking across the genome as sort of training examples rather than across individuals. And I'm just listing here a whole bunch of models that have come out recently. If I left some off, apologies, this field is moving very fast, but this is an example of the range of approaches that has been developed in this general space of sequence to function models. So existing sequence to function models do a pretty good job of identifying regulatory sequence. So identifying regions of the genome that look like they have regulatory impact on this range of different phenotypes in the multitask setting. However, several labs have shown that these models still don't very well predict gene expression across individuals. So even if they're identifying variants or regions of the genome that do have a regulatory impact, if you actually just apply them to individual genome sequence near the genes in question and predict individual gene expression, it gets the direction wrong almost as often as it gets the direction right, okay? So the first question that I'm going to talk about a little bit today is what do they get wrong? And this is in collaboration with Nyla's lab, who's here, and I think going to be moderating the rest of the session today. So we started talking about just like, what are the possible explanations for what it's getting wrong, right? And there's simple cases like it, picks up that the lead EQTL has a function and has some impact, but it just predicts the sign incorrectly. But there's all sorts of other possible complications, such as confusion with nearby variants in LD, nonlinearity in these models as it combines multiple variants together, and potentially this long tail of rare variants that are also present in the individual genome. So first of all, just as a note, the rest of this analysis in this small section is going to be performed in the MAGE large RNA-seq data set that came out of Rajiv McCoy's lab recently. So this is a multi-ancestry analysis of gene expression over 731 individuals with LCLs. So we recapitulate the results that multiple labs have shown now of this confusion of predicting individual expression. We used Informer here to predict individual expression for 12,000 genes over these 731 individuals. And every point here is assessing the correlation of the predicted expression with true expression. So every point is a gene. And many of the genes have significant correlation. I'm focusing now on genes that have a correlation not only that's significant, but is a magnitude above 0.25. Of these genes that are highly correlated and well predicted, a little more than half, so actually about 2/3 of the genes are positively correlated and 107 are negatively correlated. So we do observe that sort of sign flipping here as well. Then the first thing I want to raise is that, like, is there a discrepancy between the reported metrics in a lot of these sequence to function models like Informer and Alpha Genome that say they do a really good job of predicting which variants are going to be EQTLs, and the personal genomics results that I just showed and that have been shown previously by other labs? And I guess the answer is, they didn't do anything wrong, right? The published metrics, this is in the same data set with our exact same predictions and analysis. The published metrics simply collapse accuracy in a different way. It's a different task, right? There's also a question of whether the focus on highly confident fine mapped variants that most of them do filter their results for first, does that make the task easier? Which is like a pretty natural hypothesis to have. If you focus only on highly confident fine mapped variants, which you would only know if you ran an EQTL test, does that make the problem much easier? And I think the first thing is, to my surprise, like, not that much easier is the answer. So here I've segmented genes by the lead variant having high confidence fine mapping, so a posterior probability of inclusion of greater than 0.9 or less than 0.9. And you can see the proportion of getting the sign wrong is like almost the same. And this is still true as I bucket down for lower PIP as well. So that doesn't seem to make the problem like that much easier for these tasks. However, it does clarify which wrong results are simple and which ones are complicated. So if you look at the cases where the predicted expression has the correct sign versus the negative sign, the flipping or the incorrect prediction of the sign of the lead variant explains a lot of it. So that means that incorrectly predicted effect direction of the lead variant does explain many of the genes that have the wrong sign. Okay, so that's what we see here. The left-hand plot is high, like confident fine mapping genes, and the right is not as confident, like low PIP fine mapping genes. So when we have a low, like a not very confident fine mapping, the flipping of the lead variant doesn't really predict whether the gene is going to flip. But when you have a high confidence fine mapped variant, the flipping of the top variant does tell you a lot about whether the gene entirely is going to be flipped. And just to give examples of what that looks like, so this is an example of a very simple and very well predicted gene from Informer. So this is a gene that appears to be controlled by a single very strong EQTL, so likely the causal variant. Informer gets the variant correct, and it gets the direction of effect correct, and it mostly thinks all the other variants don't really matter. And so it predicts this gene very well with a correlation of 0.84. And the only variant that really matters is the one that's probably causal. Likewise, we see simple flipped genes, perhaps not quite as simple in terms of identifying the causal variant, but still a very high PIP here. It kind of gets the right causal variant, maybe, but it predicts the direction of effect wrong, right? So we can see the predicted effect direction is positive. The true EQTL beta is negative, so it gets the whole gene wrong. Nyla and others have shown that this does vary with the proximity to the transcription start site, perhaps distinguishing a bit between like promoter variants and enhancer variants and the connection between enhancers and genes. But they have shown that this ability to predict the right sign for the lead variant does fall off with distance. But one thing that I want to highlight is that lots of genes don't have this simple architecture, right? So for genes with complex LD, many potential causal variants, ambiguous fine mapping, even with highly confident predictions and very strong EQTL p-values, many variants are playing a role in prediction. Potentially many variants are playing a role causally. And we get a much more complex case, right? So some of the lead variants, in fact, the lead variant is predicted with an accurate sign, but there's enough others that are either in LD with that variant or perhaps even other causal signals that don't agree that the overall correlation for this gene is negative 0.65, so highly negative, even though it's getting the main variant correct. So on top of getting some variants potentially wrong, in this case, nonlinearity is also compounding the errors. So if we take the effect sizes predicted by the model in terms of in silico mutagenesis, and instead of using the model to overall give us a prediction for the gene, we linearize it by just taking a sum over the genotypes for each individual weighted by their ISF-derived variant effect sizes, so creating just a linear model out of that. In this case, it actually then gets the right sign, so it actually predicts the gene positively correlated with prediction instead of negatively correlated. And we see a large number of genes actually that improve using these linearized models. So something like 70% of sign flip genes improve when using this linearization, which means it's not just that there's lots of variants and it might get one or two wrong, but it's potentially also combining them wrong, right? So this is just a visualization that in the case of sign flip gene, lots of them are improved with linearization. And finally, at least for the sign flipping cases, these complex genes are not being driven, the incorrect prediction is not being driven by rare variants. And we can see this simply by masking the rare variants upon prediction, and that does not rescue these genes, in fact, often makes them worse. So the rare variants are not what is causing the sign flipping errors. That's not to say that we shouldn't account for rare variants or that that doesn't drive accuracy at all, but it doesn't really drive the sign flips. Okay, so I think that our hypotheses about what these sequence to function models might be getting wrong is kind of in order here, that often the lead EQTL effect is just predicted incorrectly. And that's something that we can work on. But we also have multiple causal effects, some in LD, some multiple causal effects, combining to provide these incorrect predictions in some cases. So I think all of these are things that we can think about as we work on how to improve these models. So one direction that I'll tell you about briefly is fine-tuning models based on personal genomes, right? So if the problem with some of these models as they stand is that they've just never really seen genetic variation, they've been trained on the reference genome across loci and across contexts, but they were not trained actually on individual genetic variation. So labs here, including Katie Pollard's, Nyla's, and Sarah's, have shown that by showing these models genetic variation combined with functional data across 500 to 1,000 individuals, you can start to generalize better to new individuals. So you can build models that for a new person can predict their gene expression for genes that they have seen before, but really not so accurately for genes that the models have never seen before, raising the question of like, are they memorizing variants for those particular genes? What is going on? So they're really not generalizing well. You can see in this green bar here, in fact, sometimes worse than before fine-tuning on personal genomes. So I did list the sample size for the studies published here, again, ranging from around 500 to around 1,000. So one question that immediately comes to mind is, would larger sample sizes with more individuals and more variation help us? So we decided to go ahead and see if a much larger sample size made a difference. So the UK Biobank proteomics data offers one of the largest resources of paired whole genome sequencing with functional measurements, giving us around 50,000 individuals with whole genome sequencing and plasma proteomics. And this is a well-powered study able to find lots and lots of pQTLs, so protein QTLs. And of course, I acknowledge protein is different than RNA. The models were largely not largely not pre-trained using protein, but we see a lot of the same patterns. So I'm kind of going to gloss over the proteomics question for the rest of this. Okay, so, again, a bigger sample size is one thing, but like, why might that help? So a large whole genome sequencing cohort provides lots more genetic variants that might help us fine-tune these deep learning models. So if you think about the number of variants that you observe per gene, in different sample sizes of individuals, ranging from 100 to 1,000, 10,000, etc., the number of variants that you come across, and particularly the number of rare variants that you come across, grows quite a lot, right? So as a sample size of 1,000, we've seen nowhere close to the range of rare variants that we might observe for these genes. And as we're getting up to 50,000, right, we're accessing much more of that space. So we did take, in this case, the Borzoi model and fine-tune it on UK Biobank proteomic data across individuals. This is going to be held out individuals for observed genes to start with. And just to note, like others, we also add not only trying to predict the overall proteomic level well, but also add this sort of contrastive term, which seems to make a big difference in highlighting the difference between individuals as we fine-tune it to predict protein levels. So the first thing to note is that fine-tuning per gene does, as others have shown, improve prediction on held out individuals, and actually, maybe somewhat novel, observe that the fine-tuned models do in many cases perform a little bit better than a linear model like an elastic net model or others. And we're exploring, are they picking up nonlinearities with some evidence that that is the case, so that some genes might actually benefit from nonlinearity in their prediction. And we can see how sample size is affecting that now, right? So previous studies, again, being somewhere between 500 and 1,000, we do see that on this per gene prediction task of held out individual protein levels from personal genome sequence, that we do see an improvement, going from 500 to 1,000 is certainly a big step, but we're seeing improvement up to 5,000 and we think beyond as well. So the sample size is helping even with this task. Furthermore, it is definitely picking up and prioritizing rare variants among the high-scoring positions based on in silico mutagenesis. So here comparing our fine-tuned Borzoi model to an elastic net model, looking over a minor allele frequency, a large fraction of the high-scoring variants for Borzoi, for the fine-tuned Borzoi, are in fact rare in comparison to what elastic net is able to pick up. And again, noting that this elastic net model was trained on this large sample size, where it has some power to detect the effects of rare variants. We are still picking up much more. We have also shown that masking the rare variants does degrade performance. So I think there's a combination of nonlinearity and rare variants that we're capturing now that other models were not able to before fine-tuning or with just a simple linear model. Okay, but the next question is, does this fine-tuning across both genes and individuals allow us to start predicting effects on unseen genes? And one thing I'll note here is that we actually only define the loss in terms of the pairwise contrastive effects. We no longer, due to how we handle covariance and normalization, even really try to predict the absolute protein expression level. We only look at the difference between individuals. And what we see here is that as we increase our sample size, both in terms of the number of individuals and the number of genes we're able to see, we go from hurting the performance of Informer, this is Informer, apologies, we've now switched to Borzoi, from hurting the performance of Informer and other models to actually starting to see a reliable improvement. And while it's not large, we do see this extend across, multiple epochs and lots of different runs of the data, suggesting that this is actually a robust, if small, effect in improving our ability to generalize to new genes. So from this, I guess we'll sort of summarize this part of the talk. Do we need machine learning and AI for this task? So I think if we really want to be able to extend to rare variants, yes. I do think we're also capturing some nonlinearity in these models. So I think, again, there is power here. But we still have a lot of errors that we can improve. So I think we can still improve on rare variants. We can see we have room to go on common variant effect direction, and especially in combining multiple variants across a full personal genome. So do we have the right data? I think this is very much an open question. So the much larger sample size of UK Biobank, although it is proteomics, does improve our model accuracy. It does start to improve multi-gene fine-tuning, but I think there's still quite a long way to go. And the data covered by the space of just this natural variation may not be enough. So I think the field can think hard about like what data would really support this task. Okay, so I'm not exactly sure what time I should end now, given the delay, but
I think we started 20 minutes late, so I think we should try to wrap this up at 10:30, right? I know that.
can do that.
Yeah.
Okay, so I'm going to totally switch gears now and talk about predictions from functional omic data. And this task is arose out of an idea for characterizing environment-related disease risk prediction. So we know that the environment impacts us, in many cases for some diseases as much or more than genetics. And environmental and behavioral effects like diet, alcohol, nicotine product use are a major public health burden, accounting for, hundreds and hundreds of thousands of deaths per year. And more so, these diseases can present after many years of exposure, at which point they can be quite hard to treat. So early detection or early risk assessment could really make a difference in terms of public health. And there's pretty big challenges in the current clinical assessment of individual environment-associated disease risk, right? So personal risk varies even with the same exposure level. So, right, people hear many people talk about their aunt who smoked her whole life and was fine, right? Individual biology, genetics, background health, other environmental effects, and age interact with the exposure. So it's, it's different from person to person. And furthermore, patients are not always forthcoming about their habits, environment, and lifestyle risk, right? If a doctor asks a patient, how many cigarettes do you smoke per week? My clinician friends will tell me not everyone is honest in their answer. Both of these factors mean that self-report is an imperfect predictor of disease risk. So here we wondered, can we use omic data to assess how much risk somebody may have actually accrued from these types of environmental exposures? So here we're going to use plasma proteomic data to learn signatures of environmental exposure, and using those proteomic signatures of the environmental factor, base a predictor of disease risk on that, right? And then we'll go back and look at the models and what they're telling us. But essentially, we want to see if we can tell how much risk somebody has accrued from these types of environmental factors. And again, here, I promise my lab doesn't only work with UK Biobank, but this is again going to be UK Biobank, where in addition to proteomic data from plasma, we also have diverse phenotype information and behavioral and environment annotations for our individuals. So we developed a method that I call the PANDA score. So this is proteomic augmented neural network damage accumulation score, a mouthful. It is a machine learning based plasma proteomic signature of environmental exposure that we can then use to predict exposure related disease risk. So why might this be interesting? So the plasma proteome, of course, captures proteins from many organs and systems across the body in an accessible tissue sample, and could reflect accrued changes across the body from these environmental risks. Furthermore, individuals who deviate from their self-reported or known exposure in their PANDA risk score may be of interest for developing therapeutics or disease understanding of why they might be, for example, not accruing as much risk as someone else who, say, smoked the same amount as them. So we've explored two phenotypes so far. One is connected to smoking, so pack years smoked, which is predefined in the UK Biobank and basically aggregates across somebody's lifetime how much they've actually smoked over time. We've also looked at HbA1c, which is a measure of exposure to high blood sugar aggregated over not an entire lifetime, but certainly more than a blood sugar measurement in the moment, so months. We're investigating other types of environmental exposures such as pollution, alcohol, and others, but these are the two that I will be focusing on for the results presented here. This also opened up methodological questions for us. So one of them being, how do we incorporate biological domain knowledge, right? So neural nets across many domains have been shown to be quite successful, very large models sort of once there's enough data. But they also take advantage of things like invariances in the domain, right? Like rotational invariance in computer vision, et cetera. And especially when data limited, potentially domain knowledge. So our question here is, will incorporating domain knowledge improve the application of neural networks in this disease prediction task? So here I'm going to focus entirely on using gene sets and pathways as prior knowledge of which genes are working together and use those to guide the neural nets that we'll be talking about. So we can start with just a standard neural network predictor of phenotype from proteome. And again, here my phenotype would be, for example, pack years smoked, or the blood sugar measure that I mentioned. And we're starting with the full proteome, so several thousand proteins from a single individual, and we can build a predictor ranging from a linear model to a standard neural network, with various architecture to make that prediction. Again, in this example, of course, accounting for various covariates of this data. One way that we might think about incorporating prior knowledge is to just change the connectivity of a standard feed-forward neural network. So for example, in the first layer, we might imagine aggregating and connecting proteins based on their participation or membership in a set of known gene sets. So this is a structured feed-forward neural network design. So that's one architecture that I will talk about. And the other that's a natural incorporation of graph structured or pathway structured information is to use a graph neural network model architecture, where we incorporate information about known protein sets to derive a set of embeddings to input to then a feed-forward neural net. So the first thing that we observe is that both of these structures, both the graph neural network and the feed-forward neural network, do improve by incorporating prior knowledge over what I've shown here is the same input Go gene sets permuted. So both of them do improve, but the graph neural net is clearly superior. And you may already be noticing that even with permuted gene sets, the graph neural net, interestingly, outperforms the feed-forward neural network here. And as I'm motivated, domain knowledge is improving performance, particularly when we're data constrained. So given a quarter or half of the available training data in this UK Biobank application, the improvement that we get by incorporating the true graph structure as opposed to a permuted graph structure is much more notable when we have limited data, and starts to fade as we get towards the sample size available in UK Biobank. We thought, well, we know a lot more about genes than just like one set of Go annotations. So we started trying to incorporate a bunch more sources of information, different Go categories, other sources of pathway knowledge. And we saw in our graph neural net that this relatively quickly and dramatically hurt our performance. And we just were losing all of the gains that we had over the feed-forward network and even over linear models in this case. And we thought, well, that's sort of surprising and not so good. But I mean, in retrospect, it's pretty clear that the graph neural net was just over smoothing protein embeddings. If you start to connect every protein to every other protein through this very extensive set of gene networks, you're just smoothing the entire signal away and losing all of the signal that you had in the individual protein levels. So that was really degrading performance. So the student working on this, who I will note is an undergrad and about to apply for PhD programs, so remember the name. He had the idea of, instead of just incorporating them all in one big graph, to actually derive multiple sets of embeddings from parallel graphs. So to include not just one big graph with all of the different sources of information together, but to have parallel sources. And we've done this for multiple annotations now going forward. And again, we take the resulting embeddings from the graph propagation step and just concatenate them as input to the feed-forward component of the network. And we do see that even extending from 1 to 2 sources of information here, so this is just 2 different Go categories, or 2 different types of Go annotations, for example, does give us a noticeable boost in performance. However, and again, I give credit to the undergrad for caring, to Bermuda for caring that this might be the case and testing it, is that it's not just that we're incorporating more information, it's also just simply the expressivity of the model. So we're looking at multiple different views of how proteins might be combined together. And actually, that improvement in performance tracks even when it's random. So if you add other random gene sets as parallel graphs in the graph neural network, you also get an improvement in performance. It's not as good as the true structure, and it does level off before the performance of the true structure, but that, additional expressivity is actually a big source of improvement as well. So that definitely needs to be noted. Okay, so that kind of is our initial exploration of how prior knowledge in its sort of simplest form, just Go gene sets and other pathway information, can be incorporated to improve performance on this task. But it really was motivated by a true clinical need. This is in collaboration with a pulmonology colleague at Hopkins. We really did want to see if this PANDA smoking score, so predicting pack years smoked by these individuals, could help improve our prediction of individual disease risk. So we took our PANDA score that was trained for pack years smoked. So, we trained on we trained our initial predictor on pack years smoked. And then we wanted to see on a held out set of individuals, so not included in the PANDA training at all, if we compute their PANDA scores and use it to try to predict smoking related diseases. Here I will show you COPD, but we have basically similar results for high blood pressure and a range of other smoking related diseases here. We actually just use that very simple single score to try to predict disease risk along with covariates such as age and etc. How does that compare to if we just took the annotated pack years smoked directly and use that to predict disease? And UK Biobank, I will note, like the self-report, I think is probably more accurate than a standard self-report you would get in a clinic of pack years smoked. So I think this is actually like a harder baseline than what you would face in clinic. And we used it here to try to predict who would be diagnosed with COPD. And first of all, what we see is that the PANDA smoking score does provide an overall strong predictor of COPD diagnosis in test individuals not used in training of PANDA. But more excitingly, this score predicts COPD diagnosis noticeably better than self-reported pack years itself. And it's not simply the same signal. If you combine the two, the PANDA score and self-reported pack years smoked, you get an even better predictor of who is likely to be diagnosed with COPD. And this improvement is consistent again across multiple smoking-related disorders. So we're quite excited about this and kind of in the process of looking at what the model prioritizes, which does appear to be a lot of lung-related proteins and things that look exciting and plausible. So thinking about how this could, really be, something like this could really be used to improve disease risk prediction. So our conclusions from this step is that prior knowledge can help. Introducing overly strong model constraints, such as we did in the feed-forward network, can actually be limiting in her performance. Expressivity is a major factor, regardless of prior knowledge, and that both of these interact with the data set size that's available. So how much training data is really there can kind of tell you about the trade-off between those two things. And certainly more exploration is to be done on how to incorporate domain knowledge and inductive bias in biological settings. But very excited that this PANDA score looks like it can help improve disease risk prediction over self-report of these type of exposures. So overall takeaways, like on the DNA side, I think we're still quite data limited. Biobank scale data is helping, but I think other types of data and other ways to think about improving our effective sort of range of variation that we cover may help. On the functional omics side, I think we've shown that it's worthwhile to explore how to use domain knowledge. One thing I didn't highlight in this talk, but I just always say anyway, is that confounders, especially in this functional data, really, really make a big difference. And if you don't attend to them, you will get very misleading results. And for both types of tasks, how we define our phenotype and how we define our task really can change our apparent performance and what these types of models can be used for. And I think I'll go ahead and end there and just thank the members of my lab, especially Eduarda, Jake, Prabuddha, Fuchen, and Gus, who led the analysis that I showed here, along with collaborators Nyla, Rajiv, and Meredith being the pulmonologists that we're working with on the last project. And thank you.
Yeah, we have some microphone runners somewhere, I think. Yeah. Rich, thanking you. It may or may not be on.
Yeah, so with the graph neural network stuff at the end, as you get to even a third layer in a graph neural network, GCNN, this diffusion, sort of everything becomes gray issue is an old issue. In the past, people have used skip levels and other things, and I think now that we're kind of getting into neural fields and some other things, but did you have skip level in that model?
We did not have skip levels in that model. I would say most of that is relatively new, and I think it's a great suggestion to see if that.
I mean, I like the way you
change, if we don't need to separate them then, if we change exactly how the information is propagated is a great idea.
Cool.
I want to, I guess first say it's a fantastic talk. I really learned a lot. And I guess my question is applying across both talks, but I'm also curious on whether when you're comparing these models, you found that there are complementary predictions, for instance, because I guess like when I think about nonlinear AI models, they might be predicting something as well. And I'm just curious on whether you found whether you can ensemble like a simpler, I guess, a little model with the AI models to get even better predictions in your experience.
That's a good question, and it's certainly been observed. I don't know if Nyla may have more comments even that, these models make different errors from each other, from one to the next. And so certainly ensembling them or thinking about ways to combine them perhaps with estimates of their uncertainty would be a good way to go.
One clarifying question. So the Panda score is trained on self-reported smoking, is it correct?
That's correct.
Then a philosophical question is like, how do we decide when to trust the observation versus the prediction? Because it happens in sequence to function models too. Like sometimes predicted profiles look much more reasonable than the observed data. So we tend to trust the predictions more than the observations. So I think you have something similar, but how do you decide between the two? I mean, one simple way is to predictor basically, but whichever predicts better, you trust it more basically, but.
Right. So I suppose as a disease predictor, we've shown pretty convincingly for smoking that Panda is a better predictor of disease, but it's not necessarily only because it actually is capturing their true pack years better than their self-report. We would really need a cohort where we had some way to assess like how much is this person really smoking versus what do they say? Like we don't have any sizable data of that form yet. I think perhaps like one way to start to look at this is in biological interpretation of the model, and biological interpretation of the individuals whose Panda score most deviates from their self-report. Like, do we have evidence that they actually have different underlying health conditions? For example, they may have lung inflammation for reasons aside from smoking on top of smoking, and that may be what we're partially picking up. So I think we need to look at what might explain the differences between the predicted and true score and try to interpret it if we don't have this really nice gold standard, which is certainly one of the limitations we face in all of these biological applications.
Another question. Great talk, really enjoyed it. So you have a slide where you're comparing the graph structure neural network, graph neural network, the feedforward network, which has been dropped out or by. So in that slide, your commuted gene sets for the graph network came out better than the feedforward network. I was just wondering if you could comment on that. It seems a bit counterintuitive. And does it reflect systematic difference in the network? So the setup makes for that.
Yes, I think our leading hypothesis is that we have really heavily constrained the space in the feed-forward version. And in fact, if you add in additional connections around the collapse to the gene sets, you can recover some of that. So if you just use the individual protein levels directly connected to the next layer 2, you do recover some performance, but you so I think expressivity is and like overly constraining the model is our main hypothesis. But I do have to say, like, we didn't really push on like all the ways we can optimize the feed forward network either. And so if there are like ideas that you think we should try there, I'm not trying to overall say feed forward is bad, graph good, but if there are other versions you think we should try, happy to consider that. Yeah, thanks.
Okay, great. We'll move on to the orals, see how setup goes. Yeah, we'll see. So suppose if I do this, then that fixes things. I did some chat GPTing for advice, so we'll see if that actually. Okay, so what are you seeing now? I think this is all good. Oh, I need to be screen sharing. So.
Is it just the one mic at the back?
Is it the full slideshow with animations?
Yes.
Oh wow.
Yes. Yeah. Various tricks. Okay.
I think we only have one mic working at the moment, so I'm gonna
try to moderate from here. So yeah, after Alexis's great talk, we're excited to have another set of short talks. We try to keep them on time since we're already a little bit late and stick to the kind of 20 minutes each. So I'll try to give you a few minute warning. Okay, so first talk is from Johannes Linder, and it's on joint prediction of mRNA expression, degradation rates, and RPP binding events. And take it away.
Thank you. All right, so I'm Johannes. I'm excited to be here to share some preliminary findings on the potential performance improvement of genomic sequence prediction models when augmenting their training data with various post-transcriptional assays. So this is my disclosure. So as kind of a general introduction to the problem of genetic variant effect interpretation, the human genome consists of more than 3 billion base pairs. The vast majority of these nucleotides do not actually code for a particular protein. Instead, these nucleotides make up a complex sequence regulatory code that governs gene expression through multiple layers of regulation. At the very bottom layer, at the layer of chromatin, you have enhancer and repressor elements spread out throughout the genome that basically control the transcriptional activity of genes. As a gene is transcribed into pre-mRNA, it's further subjected to additional processes, including splicing and polymorphisation, and these processes are themselves governed to some extent by a sequence code embedded in the pre-mRNA molecule. Then this mature transcript can now go on and become protein product, and along that journey, it's again further subjected to additional processes such as mRNA degradation or control mediated by translation efficiency, and again these processes are to some extent governed by a sequence code embedded in the mRNA molecule, both in the coding and non-coding regions. And it's well known by now that numerous non-coding mutations actually cause or contribute to various genetic diseases simply by disrupting these various regulatory functions. And I want to emphasize here that sort of we know about the deleterious mutations that really occur at each of these regulatory steps. So it's very important that we can develop accurate models that allow us to actually predict the functional impact of genetic variation with respect to all of these regulatory phenomena, right? And that's a very daunting challenge. And one sort of flexible and to some extent pretty powerful approach for predicting or for jointly modeling many of these various regulatory functions is to reduce the problem to that of predicting various genomic sequencing coverage profiles, like one-dimensional coverage profiles over the genome, given only the sequence as input. That was sort of an approach popularized by the very successful Informer model, and before that introduced by the Senji models. In David Kelly's group at Calico, we introduced this other model called Borsoi somewhat recently, where we kind of took that step, we took that idea one step further, where the idea now is to learn to predict raw tissue and cell type specific RNA-seq coverage profiles from DNA sequence alone. The intuition there being that if you can teach a parametric model to actually predict RNA-seq coverage patterns from sequence alone, it must have implicitly learned about many of these various regulatory steps that I talked about in the previous slide, including transcription activity and splicing and polymorphisation patterns. And there are many different ways of evaluating these models, of course, but it turns out that these models can quite accurately predict, for example, held out gene expression values of sequences not seen during training if you sum up the total predicted coverage over the exons of a gene. But one blind spot, if you will, of the Borsoi model was that it wasn't really trained on particularly many, or for that matter, any post-transcriptional assays. So assays that measure many of these post-transcriptional regulatory phenomena. And if we look at the actual functional landscape of what goes on after transcription, we have a lot of different processes that can, act and interact together. For example, you have RBP proteins that come in and bind to cognate motifs in, for example, the 3' UTR of an mRNA molecule. These RBP binding sites can be interspersed with polymorphisation sites, and these two functions can, of course, affect each other. Namely, RBP motifs can improve the efficiency of cleavage and polymorphisation, and in turn, polymorphisation sites can implicitly dictate which RBP binding sites will be included in the mature transcript. And finally, of course, some of these RBPs are actually mediating RNA decay by by recruiting degradation components. So these processes are clearly very interconnected. But if we look at the current sort of landscape of machine learning models that are used to predict the outcome or predict these various post-transcriptional functions, you typically have these very specialized models that are trained on bespoke data to predict each of these functions. So for example, to predict mRNA degradation rates, there's the Saluki model from Vikram Agrawal and David Kelly. There's more recent work, for example, the Orthros model that can seemingly more accurately predict mRNA degradation. To predict alternative polymorphism, there are various types of specialized models. And then, of course, there are many different kinds of specialized models to predict RBP binding events. And, I will say all of these models are very accurate and good at what they do. But here, in this sort of smaller study, we asked whether a model trained to jointly model many of these regulatory processes in sort of one parametric model would improve its generalization capabilities. In particular, we asked the question whether RNA-seq coverage prediction improves if you jointly model post-transcriptional assays as well. And this work is sort of inspired by the really great big RNA paper from Deep Genomics, where they clearly showed that an RNA-seq coverage predictor that is fine-tuned to predict clip profiles can do that very accurately. So here we ask kind of a tangential and related question, namely, if you pre-train the model to predict post-transcriptional assays in addition to RNA-seq, does the RNA-seq coverage prediction quality improve? And as a nice bonus, you then also get a model that can now score genetic variant effects with respect to all these different regulatory functions. So to do that, we developed this kind of smallish model, in today's terms, at least. It's a 40 million parameter model. It's somewhat smaller than the Borsoi model because it's trained on a little bit less data. In this kind of preliminary work, we call the model Bursuki as kind of an homage to the Saluki model, which was trained to regress scalar mRNA decay rates. So we feel that this model now tries to sort of combine Borsoi with the Saluki model. So basically, we collected data covering 8 human and 4 mouse cell types. First of all, we trained the Bursuki model on a subset of the data that we trained the original Borsoi model on, that we feel are good training data whenever you want to teach a neural network model about transcriptional regulation. This includes, ENCODE DNase and RNA-seq, as well as phantom cage profiles. But now we're including these additional post-transcriptional modalities that capture one form or another of post-transcriptional regulation. For example, we included data, basically some really nice quality single-cell perturb-seq from Rahul Satia's group here at the Genome Center, actually, of basically performing perturbations to individual polymorphism cleavage regulators and reading out the transcriptome with 3' single-cell RNA-seq. So we trained the model to predict these pseudobulk perturbation coverage tracks. Now we're also including coverage profiles of ENCODE eClip profiles, as well as some really nice currently unpublished clip data from the Gudarsi lab at UCSF and the Arc Institute. And then finally, we collected some of the mRNA degradation data sets that the Saluki model was trained to predict. Now, the Saluki model was trained to predict these mRNA degradation rates sort of as a scalar regression task. What we're doing here is a little bit differently because we want to move this data into this joint framework of predicting coverage profiles. So what we're doing is that we're actually generating a bigwig coverage track for each time point in an mRNA degradation data set, for example, a transcriptional shutoff experiment, and tasking the model with, in a multitask mode, to learn to predict each of these time points and their coverage profiles, which may make it a little bit more powerful than scalar regression, seeing as you can now observe differences, relative differences across time points over different isoforms, for example. So after having trained the Bursuki model on all its data, the model can seemingly predict RNA-seq coverage within these mRNA degradation tracks, as well as clip profiles quite accurately on held out test genes that weren't seen during training. And one conclusion really is that once you include all these post-transcriptional assays in the pre-training data, the model actually improves its test set performance on the RNA-seq coverage prediction tasks. So seemingly you're getting a higher quality predictor from that. And furthermore, the model can quite accurately predict both the total magnitude and relative coverage profile of these CLIP profiles as well, or CLIP experiments. Another interesting vignette is that you can now actually estimate mRNA decay rates from these coverage tracks. So you kind of retain many of the powers that you get from scalar mRNA decay regression. So basically what you can do is you can just predict the RNA-seq coverage over genes for an mRNA degradation time course, and then sum the total coverage of RNA-seq coverage over the gene. And then with these scalar gene expression values, you can fit a decay curve to obtain an estimated mRNA half-life. And you can now compare your predicted mRNA half-life to the measured mRNA half-lives, and you observe quite good concordance on held-out test data. And then finally, it's quite nice now that we have this model that basically allows us to interpret the impact of genetic variation with respect to many different regulatory phenomena, namely, steady state expression, mRNA decay rates, RBP binding, and other modalities. So to illustrate what that might look like, here I'm showing an example where we're making coverage predictions over the PDXP gene, which is a quite interesting gene because it harbors a conserved repressive ARE binding motif in its 3' UTR. And so I'm showing you the wild type coverage predictions over an mRNA half-life time course experiment, as well as CLIP profiles for E-level 1. I'm showing the wild type predictions in blue, and then overlaid in red, I'm showing you the coverage prediction, average coverage prediction, as we are ablating this ARE binding motif by replacing the nucleotides overlapping that motif with effectively random nucleotides. And so, as you can see, the total predicted RNA-seq coverage increases in all of the time points, which is what you would expect when you're now removing this repressive motif. But then we're also seeing this relative increase in fold change over the later time point as opposed to the first time point, which basically implies that the model now predicts that removing this motif actually results in a more stable RNA transcripts, and which is also what you would expect, because ARE binding motifs basically recruit the degradation complex. And then finally, of course, at the bottom, you can see that we're now predicting loss of E-level 1 binding by removing this motif. And so we evaluated the Borsuk model, beyond just looking at one example through a number of different ways. One thing we did is that we collected or downloaded this publicly available MPRA data set that measured the effect of genetic variation on mRNA half-life. And we inserted these MPRA constructs into the endogenous 3' UTRs of a bunch of randomly chosen genes and sort of calculated a marginalized variant effect score. Unfortunately, with the 4-fold ensemble of the Borsuk model, those variant effect predictions did not beat the 50-fold ensemble of the Saluki model. Saluki is much more computationally efficient, so you can ensemble much more there. But a small win is that the variant effect predictions of Borsuk combined with Saluki is better than any model alone. So that's something. And then if we start looking at genetic variants in an endogenous context, for example, if we want to classify putative causal Mendelian variants from the trait data set, relative to match negatives, the Borsuk model actually does quite a lot better than the Saluki model for genetic variants specifically located in the 3' UTR. And furthermore, the Borsuk model trained on all of the post-transcriptional assays outperform sort of this controlled Borsuk model trained on only the baseline data. And you're seeing the same trend at the task of classifying fine map causal eQTL SNPs from matched negatives. So in summary, Borsuk is trained to jointly predict RBP binding, expression, and mRNA decay rates. And these preliminary findings suggest that this joint modeling approach improves test set performance for RNA-seq coverage predictions, as well as a higher quality variant effect predictor model. And I want to end by just acknowledging, David Kelly, who mentored me on this project, as well as all the fantastic lab members I get to work with in Kelly Lab at Calico Life Sciences. And also a big shout out to the Godarcy Lab for sharing their really excellent clip data with us to train on. Okay, so I'm happy to take questions.
Thanks for the interesting talk, Johannes. I wonder how does the model apply to cell type specificity or what cell type of cell lines are the model?
Yeah, that's a great question. We didn't really evaluate, so far we haven't evaluated the model in terms of finding at the task of identifying cell type specific elements related to mRNA degradation, in part because the data sets that we trained on are kind of, these basic cell lines that so far studies have kind of shown don't harbor too many cell type specific effects in terms of degradation. If we're expanding this to more interesting cell types, I think that would be excellent to do.
Thank you.
Hi, thanks for the interesting talk. Have you tried just providing the model with the splice sites? The reason I ask this is like, the encode eclipse data is not great. Probably about a third of it is not sequence specific. And the proteins that aren't sequence specific normally just mark splice sites. And one of the best predictors of mRNA half-life is number of exons. So one wonders whether or not these are just surrogates for splice sites. Certainly predicting splice sites would give you a very good model of post-transcriptional regulation.
Yeah, that's an excellent comment. And the previous papers that have trained other models to predict these data sets have indeed shown that the number of splice sites in a splice transcript is an excellent predictor of half-life, is a good predictor of half-life from these data sets. But they also showed that beyond that as a baseline, these sequence models tend to perform better. So there's probably some residual benefits from sequence modeling. And there are things then that we are gaining with a sequence model, like the last example, we're actually identifying ARIE binding motifs or familial binding sites, things like that, is an orthogonal axis of mRNA decay.
Hi. So in the earlier slides, you mentioned that does the joint model improve anything? And you mentioned that RNA-seq predictions benefit from other RNA properties. But is it true also for the other way around when you compare a single class model? And second question is more technical. If you look at the RNA properties and contributions for the DNA signal, I assume that you don't have any explicit masking for the DNA features when you make the RNA property predictions. Do you see anything interesting there? Or does it make sense to even use the DNA sequences for prediction?
For the first question, we did not look at sort of the other direction, if whether the RNA-seq coverage predictions lends any benefit to the other assays. We only did one ablation where we basically removed all the post-transcriptional assays, so we could only do the comparison in one direction, unfortunately. I think that's an excellent suggestion. And to some extent, the big RNA model in the Deep Genomics paper showed quite clearly that the RNA-seq coverage predictor, you can fine-tune that to predict clip profiles really nicely. So that's pointing in that direction. Could you say your second question again?
So using the DNA sequence like beyond the RNA boundaries, basically for like and half-life.
I see.
Right. Like of these sequences, or
no, it's the full sequence, which is kind of needed because you're also jointly trying to learn to predict steady state expression levels.
Have you checked attributions in these regions?
No, we haven't. It's an interesting thing to look at for sure. Yeah.
Okay, great. Yeah, thank you.
Correct.
See how setup goes for you. And you need to share on Zoom. Yeah, that's it. So that's good up there.
We have all the middle one.
Okay, awesome. Yes, perfect.
Okay, cool. So, yeah, the next talk is going to be presented by Alan Murphy and Masih Nagai, and it's on mitigating catastrophic forgetting in genomic foundation models with continual error. Go ahead.
Thank you. So I'm Alan. I'm a postdoc in the Peter Cruz group at Coastal and Harbour Labs. And along with Moon, we're going to present our work in progress on mitigating catastrophic forgetting in genomic foundation models with continual learning. So to kind of set the scene, although this may not be totally necessary given the group here, we're interested in sequence to function models, which are neural networks that take in one hot encoded DNA sequence and predict some activity like transcription factor binding in the cell type, or multiple activities in a multitask setting. These models have been broadly applied to understand the functional consequences of genetic variants and analyze cis-regulatory logic. Moreover, these multitask generalist models have been used as foundation models for downstream tasks through fine-tuning. However, despite recent advancements, more recent work has showed that these models are still poor predictors of genetic variant and cis-regulatory effects. So you'll recognize the plot on your left from earlier on, where from the Mustafavi lab, they showed that on personalized genomes, that is matched DNA sequence and expression from individuals, the y-axis shows that informer poorly correlated with true individuals' gene expression. On the right-hand side, based on CRISPR-I validated enhancers, the Gagne lab showed that the true knockdown expression in red did not match informer's predictions in blue. So why is this? If we consider the current training process that all these models follow, they all use a single reference genome to predict thousands of different activities. So in a sense, they're learning very broad statistical association between DNA sequence and activity. So we believe these issues are really symptoms of this current training regime, whereby models do not learn causal mechanisms and instead just learn broad correlations. So how can we correct for this? One option is to expose the models to more genetic variation at training time. Two choices of genetic variation are personalized genomes, which we've heard about already, and perturbation assays like multiplex assays of variant effects and CRISPR interference screens. So first, let's look into the details of personalized genomes a little bit more. So this scenario of research that's really exploded over the last 2 years or so, and the general approach is to take a model that's been trained genome-wide like Informer, and then fine-tuning on personalized genomes. However, most of these models fail to significantly outperform linear baseline. Here we're showing the results for one such model, where in the center you see its performance on a held-out population. To the left is the performance of the model before fine-tuning, and to the right is the performance of the linear model. So why do these fail to outperform these linear models? One possible issue is that there's just insufficient genomes. We heard already about scaling this up to 50,000 genomes, but will this actually improve the models to significantly outperform linear baselines is still an open question. Moreover, there's differing modeling choices we could use, and I don't think that's been fully saturated either, so that could improve things. However, for us, we believe that there may just be insufficient variation across populations in personalized genomes, capture complex cis-regulatory logic. So how can we actually look to do this? We believe the best approach is to use perturbation assays like Maves and CRISPR-Is to actually capture this cis-regulatory logic. So if you consider the current approach with the reference genome and these genome-wide activity signals, you could incorporate perturbation screens like Maves and CRISPR-Is here. So the Maves will tell you dense perturbation information within a cree, whereas CRISPR-I data will tell you the distal effects of crees. So these dense perturbations they inform locus-specific causal mechanisms. However, learning at a locus does not ensure generalization. Another issue is that there's currently no framework to incorporate this locus-specific perturbation data. You could think to multitask these two data types, but that would result in an imbalanced data set, where at certain locus in the genome, you have a lot of data, and then at the rest, it's very sparse. So for this, we're proposing our causal refinement framework to incorporate causality into genomic deep learning models. The first step is, as usual, you pre-train a model genome-wide with a reference genome in a multitask setting. Thereafter, though, you enter this causal refinement loop, conducting locus-specific iterative fine-tuning to refine the model's understanding of cis-regulatory logic. This loop allows us to add new perturbation experiments as they're released, or even use the model to inform the next best perturbation experiment to include through an active learning framework. However, there is an issue in that iterative fine-tuning has been noted to lead to catastrophic forgetting in the general machine learning field. If this is the case, it could hamper our model's refinement of the cis-regulatory logic in this loop. So we set out to investigate whether catastrophic forgetting occurs in genomics and to what extent. So to do this, we went back to these models that fine-tuned and form our own personalized genomes. We first looked at the models from the Immunitas group, who used something similar to the Siamese neural network architecture and produced multiple models. Here we're showing the performance of these models on the original informer task, whether that be across all tasks informer predicted or specifically in case transcription initiation. And what I hope you can appreciate is all of these models forget or do worse on this original task after fine-tuning. And this is only after one fine-tuning iteration. We plan to do this iteratively many times, so we wanted to investigate what would happen in that setting. And also we wanted to test another training approach that's been used in the field. So we looked at the regularization loss function by the Pollard group, whereby they basically regularize for differences across DNA sequences to basically improve performance on these personalized genomes. We then took the GVitas data set and split it into 10 training groups, and then iteratively fine-tuned on these groups one at a time. Here I'm showing the performance on each of these 10 groups after fine-tuning and before fine-tuning a zero-shot setting. So you can see that fine-tuning does improve these models. However, when we look at the performance on the original task, you can see that the model forgets very quickly on this original informer test set. And this leads to really catastrophic forgetting. So this really is an issue for our proposed framework because this could occur when native fine-tuning.
To deal with catastrophic forgetting in our causal refinement framework, we propose to apply continual learning. Continual learning is a learning approach that sequentially trains a model for new tasks while trying to preserve previously learned knowledge. For genomics, the goal is to retain what the model learns from large-scale genome-wide profiling data while refining its causal understanding, moving beyond associations to causation. Unlike typical continual learning, which focuses on adding new tasks, our setting aims to refine causal mechanisms within the same task domain. This schematic shows a simplified parameter space. The gray region represents good performance on task 1. Starting from random initialization t to 0, the initial training moves the model into this gray region. When we introduce task 2, it has its own optimal region. Naive fine-tuning provides no incentive to remain in this task 1 region, so the model may shift to parameters that fit task 2 but lose task 1 performance. Adding task 3 can push it even farther away. With continual learning, the goal is to guide optimizations toward parameters that remain effective across all tasks, preserving prior knowledge while integrating new ones. For genomics model, this has a natural biological analogy. Many regulatory mechanisms are local-specific, but after encountering them multiple times, the model can potentially begin to recognize patterns and generalize to new loci. This is called forward transfer of knowledge. At the same time, as the model incorporates new loci, its understanding of previously learned loci can improve. This is backward transfer of knowledge. In our study, we first looked at 2 major continual learning approaches. The first one is replay-based approach. In naive fine-tuning, once the model moves to a new task, it never sees examples from earlier ones. This risks forgetting. Replay tries to overcome this by periodically revisiting past examples so the model can remember prior tasks while adapting to new ones. The second data-free approach is a regularization-based approach exemplified by elastic array consolidation, EWC. Here, the model is trained on task 1, and the importance of each parameter is estimated. During fine-tuning on a new task, deviations from important parameters are penalized according to this importance matrix. As new tasks are introduced, the importance matrix is updated. In practice, the total loss for a new task is a sum of the task-specific loss and a regularization term that penalizes deviations according to the importance matrix omega and scaled by hyperparameter lambda. The importance matrix is often approximated by the diagonal of the Fisher information matrix. We apply continual learning at 2 different scales within the Cree level and across distal Cree level. Let me start with the first setting, where we integrated Cree level made data into ProCapNet. ProCapNet is a BPNet-based model. It takes a DNA sequence as input and it predicts both profiles and accounts from ProCap peaks. These outputs correspond to transcription start site positioning and transcription initiation rates. To decide which region to build a MAVE library on, we followed a few steps. First, we picked a sequence with a high activity, meaning high counts from ProCap. Then we chose a 200 base pair sequence window based on the magnitude of attribution maps. Finally, we created a dense mutagenesis library by mutating about 15% of the wall type sequence. Now, ideally, we would like to use existing libraries or run actual experiments. But since such systematic dense libraries are not available, and due to the experimental cost, we generated an in silico MAVE library instead. An added benefit here is that we don't suffer from experimental batch effects. And for this, we used MPL Lignet as an Oracle model to predict activities of the variants. Once we had this in silico library, we fine-tuned ProCapNet on it. Then we repeat the same process again, choosing another high activity sequence, selecting a window, generating a dense MAVE library with MPL Lignet, and fine-tuning the model. We repeat this process 10 times, and we also created 10 additional libraries that were never seen during training. These were used to test forward transfer. Finally, we evaluated different continual learning strategies on 3 aspects: performance on the original ProCapNet test set, on the first library, and on the unseen libraries. Here is how performance changes on the original test set. The top panel shows the result for profiles, and the bottom is for counts. The x-axis is the number of fine-tuning iterations. What we see is that naive fine-tuning, shown in gray, causes catastrophic forgetting, especially for profile predictions. This makes sense because the MAVE data is closer to count, as in the output is a single scale activity value rather than a full profile. EWC in blue helps a little, but not enough to prevent forgetting completely. Replay, on the other hand, is much more effective at maintaining performance. Next, let's look at performance on the first MAVE library. Here we see evidence of backward transfer with the replay. Performance on the first library actually improves on fine-tuning goes. While EWC helped reducing forgetting on the original data set in the previous slide, it struggles here. And in fact, it doesn't outperform naive fine-tuning, which highlights the classic trade-off between parameter stability and flexibility. Next, we evaluate the zero-shot performance on unseen libraries relative to the original model. Continual learning, especially replay, shows consistent forward transfer of knowledge, while naive fine-tuning shows very erratic performance from round to round. In the second setting, we focused on integrating CRISPR data to capture distal Cre-Cre interactions. The setup is similar to the first setting, but here we worked across Cres. We started by picking a highly active gene with a dense Cre landscape. Then we perturbed its Cres in a CRISPR-like manner, either individually or in a combination. For this, we use Decima to predict the activity of the perturbed sequences. With the library, we fine-tune Informer. Just like before, we repeated this process 10 rounds with also 10 unseen genes for evaluation. Here are the results on the original Informer test set, with the y-axis showing Pearson R relative to the original Informer. Replay shows clear backward transfer, especially in DNA-seq ataxic tracks and chip-seq tracks. EWC also helps mitigating forgetting, though to a lesser extent. Here is a zero-shot performance on unseen libraries relative to the original model. It shows that naive fine-tuning gets worse over the original model, but we also don't see substantial forward transfer with the replay. We're looking into why this is the case, why this is different from the other MAVE setting. Might be due to the methods or how we set up this zero-shot task. EWC in blue shows an RE promise, but this stops at 5, not because we gave up, because it's actually running right now. So we'll see.
Okay, to summarize, we've introduced causal refinement as a new way to improve the generalizability and learned cis-regulatory logic in genomic deep learning models. It's worth noting that any amount of fine-tuning on genomic deep learning models can result in catastrophic forgetting. And just to note, a major limitation of this work is that we're relying on Oracle models which haven't themselves fully captured the causal mechanisms. However, this approach does give us a test bed to explore continual learning methods on a scale that would be simply too costly for wet lab experiments. Moreover, we do plan to rerun these approaches with actual wet lab experiments in the future. And just to finish, I'd like to thank everyone in the QLab past and present members and those in the CUNY and the Canvas labs for support, for helpful feedback on this work. I'll take any questions now.
Yeah, we have time for a few more things.
Okay, Dan.
Hey, thanks for the great talk. I'm wondering, so the oracles in this case are somewhat correlated with the initial models, so like Tesla and Forma, pretty related, right? Have you tried cases where either you artificially perturb the oracle prediction so that it explicitly disagrees with the original model's predictions, and see how things perform in that setting?
So for the informer model, we did explicitly pick genes where the correlation between the student and the teacher model were quite poor. So we basically predicted everything and then picked cases where they were very disparate to kind of negate that effect. Yeah, that was the issue we had early on with it.
Maybe you said this, but is it full fine tuning or it's low rank adaptation? Because there's this idea that low rank adaptation doesn't forget as catastrophically because it's a little more constrained.
Exactly.
So
It wouldn't.
with the interesting,
You still have the base model in setting. This is full fine tuning.
But even like if you do like low rank, I think that ultimate idea is that you want to update the base, so that like, in the future you have update base. But then even if the base frozen and then new ones, if they interfere and then if they forget, then you don't really get the benefit. Maybe you can just keep adaptively changing, but I think the final benefit is the updating the base.
So I was surprised that EWC did worse than my fine tuning, and I just have a clarity question where, when you have that elastic constraint, is it applied really just at the loss, which means it's probably best way to think of it is it's applied uniformly across the model. So that first question is that the case?
Yes, that would be the case.
What about uniformly?
Yeah, well, if you're just messing with the loss at the end, then the model's not really differentiating for that constraint's being applied, essentially to the whole model. But if you think about a lot of tasks where there are different things going on and the model has to learn sort of punctate different regions. So you have one model, but it's really many models involved. And just have enough breadth that you can really deal with a lot of such issues. One example is cases where you have different training style, different rotation and translation issues, different scale issues in molecular things, batch effects are another, cell types, et cetera, But a lot of that's easy for models to deal with, again, the encoding side. If you're connected to different losses, your catastrophic loss is kind of a projection, is later layers. That's my kind of another way to think of it. Is there a way to get EWC to focus on the later or the close to decode parts of the model, so that you can maybe allow it to kind of separate out into punctate regions during training? But I don't know if there are any versions of that where you sort of apply the constraint differently depending on how close to the loss you are, or where you are on that encode to late to decode space. But that'd be one suggestion, is to think about basically kind of pull that constraint over time. It seems like you solved it anyway, so maybe you don't have to think about it.
I probably don't agree that we've solved it yet, but what we've done here is we've really implemented very vanilla versions of both of these approaches, and what we plan to do in the future is to try and improve them in different ways. And looking at things like that, how much each parameter actually is allowed to be constrained would be an option. There's other issues with the vanilla elastic weight consolidation implementation anyway that we would like to try more advanced approaches first. I guess this is that starting point.
Yeah, in theory, I think the importance matrix can, capture that. That'd be great. But here we kind of approximating this, and even the original EWC, which takes Hessian, is just looking at the local curvature, which isn't amazing approximation either. So I think there hopefully would be a better approximation for genomics models.
I think it's just related to the previous question. Does land the hyperparameter that you have for the information matrix? Do you change that during training? Is there a warm-up scheme or something, or is it kept static throughout?
So the lambda for EWC?
Yes.
Yeah, no, we don't change it during training. It's currently kept static. But that could be an approach to do as well. Cool.
Okay, we should probably move on to the next one.
Okay, I have your slides, so that's good.
It will time me, or should I?
And Nyla will try and indicate, I think.
Is that fine? Is it okay if I just do a little? Yeah, whatever.
I just like the credo when I'm hitting. Yeah. That's in the.
Okay. I think this should all be good.
Okay, great.
Okay.
Okay, so our last talk before the break is Courtney Shearer talking about a genomic language model for zero-shot prediction of computer indela.
All right, hi everyone, I'm Courtney and I'm a senior graduate student in Deborah Marx's lab at Harvard Medical School. And just take a moment to thank Peter and everyone and David for this wonderful conference, and appreciate all of your patience with getting everything going this morning. It's not easy, so thank you again. But today I'll be presenting some work from my PhD where I have this genomic language model for zero-shot prediction of promoter variant effects. Maybe. There we go. So before we kind of get into the ML and everything, I like to start this talk with kind of a strong motivation. So this is not real data, don't get too excited, but this is meant mainly just for provoking some thought. The genome has a spectrum of variation, we must believe this, and kind of when I started this project, I wanted to think about where we might consider these different types of variants. Are they SNPs, indel structural variants? Are they encoding regions, regulatory regions, etc.? And we know from this release of the UK Biobank, at least for this cohort, that there is a high frequency of more than just SNP events in the genome. But where might they be? Well, we can presume that since the non-coding part of the genome is so large, they might be there. But we have an analysis from Nomad that shows us this as well, even with a fairly small amount of whole genome sequencing data from like 2020. But first, we know these variants exist, but who cares if they exist? We want to know if they do something. So we want to know if these variants are actually causing disease. So classically, people have looked at genomics, we find variants, and then we see how they affect proteomics, and we think of this as the main route to finding disease effects, which is very true, but there's a lot we still don't know. So I aim in this work to explore kind of how these disruptions in the non-coding region affect disease. Now, this is just a cartoon. There's more steps between these question marks, but we can start to think about transcriptomics for a moment. Oops. And as a simple example, and will also be, the region of most interest in our talk today, I'll zoom in on a cartoon of a promoter here, where we can imagine in the promoter region upstream of some gene of interest, we have a variant, and we have some projected change in expression levels. So what types of disruptions could we expect in these promoters? Well, in this paper, this group explored over 200,000 promoter sequences, and they took a bunch of these transcription factor binding sites and they scrambled them around, they messed with the orientation, they messed with the order, and they showed how, if we flip them around on the template, non-template strand, if we move them farther away from the TSS, we get very different expression levels just from these changes, these swaps and changes in orientation. So those are just some examples of like how we might expect, transcription factor binding sites to be very, very, subjective to context. But if we make this even simpler, these are just some simple cases I'll walk through you. We can think of case one of variants that might be upstream of the gene of interest, where we might have an indel or an insertion, but we can also think of more complex cases, maybe where we have simply more variants, we have a deletion and a SNP, we have multiple SNPs, and insertion there, and we can even maybe have like combinations of these variants in exons as well. So if we see these patterns, which we can hypothesize that they're there, how could we associate this variation with fitness effects? And if we could associate this, how would we even build a model that could find this association? And then if we had a model, just bear with me, what would we even do with it? Because who cares if you have a model if you're not going to do anything with it? So one thing we might use is we might use this model to detect deleterious variants based on some evolutionary score. If we use evolutionary sequences. We can also, if we make a generative model, we can also use this to increase power for burden testing, for finding rare variance associations. We can use such a model to improve heritability estimates, and we could even use such a model instead of just doing prediction to actually design new sequences. So we've seen that sequence models that use evolutionary information are useful for proteins. We've seen this is kind of blowing up right now. But they're still, still in our infancy in terms of using evolutionary sequences for genomic language models. But could we adopt a similar strategy just for promoters, just for today? So law-leave is an autoregressive model. It's called a control transformer because not only does it autoregress over the genomic sequence you're looking at, but it also includes these control codes where I include information from the clade, the specific, if we're looking at primates versus non-primate mammals, the species that we're looking at, and the gene that we're looking at, which I parameterize as an ESM embedding. So to build this data set, we basically took promoter regions 1K upstream of the genome sequence alignment from the Xenomia project, which was a fantastic piece of work that I believe came out near the end of 2023. And we determined our tokens for them, and we built our data set just like this. And at the end, we had, sorry, my Zoom's covering up, 13.6 million sequences. And so that's plenty of data to train a model. And in addition to grabbing our sequences from, evolution, we also took our sequences and we scored them on SAY, which is a molecular a molecular model. And it basically has a promoter score that you can leverage to see if, how promotery, I guess, the sequence you're looking at is with respect to molecular data. And we see that the red data there, our training sequences, look pretty similar to the just taking human promoters and scoring them as well. They have similar distributions. So this is a small validation that maybe our sequences are actually capturing our promoters, because again, the scheme we use to build our data set isn't based on annotations, it's just based on comparative genomics. So after this, we build this model, we introduce three benchmarks. We are going to look at EQTLs, variant prioritization from Nomad, and transcription factor binding site disruptions. And again, we're just going to be looking at indels here because we're focused on expanding beyond SNP regions. And this is kind of going to predominantly look at case 1, which I showed in that little cartoon. I'll have something after we get through the benchmarks that we can start to explore case 2 as well. But we're just talking about case 1, so single instances of a variant of an indel occurring in some individual somewhere. So for the prioritization of variants with EQTLs, it's pretty simple. Since we're looking at evolutionary models, they don't have a notion of direction of effect, but they can tell you a magnitude of effect with the absolute value of the score. And we stratified our scores for all these models using a heuristic I developed for slippage propensity, which basically just looks at where these variants are in the promoter and calculates if in that region they're in a high slippage region, meaning very repetitive, or homopolymer runs and things like this. And when we stratify by this heuristic, we can see that a lot of the models tend to do better, but lucky for us, law-leave tends to do a good bit better, and we're the best performing in this regime. Next, we're going to look at a different way of thinking about a variant effect. So we're going to look at human genetics, and we're going to look at strong evolutionary selection pressures from Nomad. Now, how we're going to do this, we're going to take all the scores that the model produce, and we're going to look at the distribution of them. And then for each model, we're going to select the lowest percentile, because what you would hope to see is if you have a variant that is very rare, the model would score it much more lowly, because in evolution, if a variant had never been seen, it would get a much lower score. So a variant that's very rare, never seen in the human population, should get a much lower score. So we're doing this by establishing some percentile threshold, and then we're just going to have a ratio of the number of variants we're seeing at that threshold, if they're the ultra rare minor allele frequency versus a common minor allele frequency. And what we see here is that, again, LaLive is consistently a top model for prioritizing these variants when we stratify by different indel lengths. But what we also see, which is cool, is we have this model called GPM promoter that's doing very well on these much smaller variants, which you would kind of expect since it's a mass language model. It's able to do very well at predicting these much smaller variants, and it's also trained on, I don't have the exact overlaps, but a much more promoter-specific data set. And then for our last benchmark, I'm going to speed up a little bit so we get to the end. I don't want to run out of time. We're going to see if models can predict context-dependent effects of transcription factor binding site disruptions. So what I did here is I took all these promoters and I fully deleted the transcription factor binding sites. And then for these two groups, we have groups of genes that have variable expression levels across GTEx and have different evolutionary pressures from OrthoDB. So we're going to categorize these promoters that are upstreams of genes that are in these two groups: consistently expressed in GTEx and very constrained, and variably expressed in GTEx and mutably constrained. And what we can see here again is that LaLive achieves the best accuracy here for this task. And we see PHILO-P doing quite well as well, because it's still a very nice model. But this is a nice way to look at promoter effect because it's combining kind of two ideas here. Now we're going to combine expression, we're going to across cell context, and we're also going to combine evolutionary constraint. So with this benchmark, we've kind of established 3 different views of promoter variant impact. We're looking at seeing if a model could do multiple things, not just be good at one thing. Can it determine if a variant is under strong negative selection? Does this variant alter gene expression? And does biological context determine disruption severity? Now, these are all the models I've shown here today are not actually sequenced to function models. These are just using raw evolutionary sequencing data. So there is probably reason to believe, maybe those sequence to function models could also be leveraged in addition to these models, but we want to be able to squeeze as much orthogonal information as we can from these different data modalities and be able to combine them to answer these questions. So not to say you should only use evolutionary sequence data, but if we have it, how can we use it? Okay. So we'll go briefly into this next small section. This is a small experiment I just did recently where we're trying to see if we can move beyond single indel events. What if we have full haplotypes? What can we do? So for this work, I got very familiar with the Genomics England data set where they have severe disorder trios, developmental disorder trios from a bunch of families. And I built all possible haplotypes from all of these families where they can have different combinations of variants in these promoters. And I also identified which genes upstream of these promoter regions are known developmental disorder genes. And ideally, what we'd want to do if we had any model, here I'm going to say LaLive, we would want the model to score potentially the variants or the promoter, the whole promoter, because remember this is autoregressive, upstream of a particular gene. If that gene is a known developmental disorder gene, we would expect it to be a bit lower if the promoter is involved, I suppose, in that gene. So we set this problem up like this. We just have a simple odds ratio where we establish some percentile threshold. And on one side of this threshold, we say, okay, how many of these genes, how many of these promoters are upstream of genes that are known disordered genes, developmental disorder genes, and over the other. And this is just a cartoon. I'll show you the real result in a second, but just to say, this might be something you would expect to see. You'd expect to see an enrichment at these lower scores, but you would hope that the models would be able to, if they're based on evolutionary sequences, would score these, rare variants, or rare haplotypes, sorry, much more low. So what we actually are seeing is that LaLive shows a 4-fold enrichment for haplotypes and known developmental disorder genes over other autoregressive models. And this is quite cool because You know, in a real clinical application, a lot of times you just have, you know, you have the raw sequencing. It's very neat if you can actually go in and just like score the whole genetic context right there and tell the patient if that whole promoter would be potentially problematic. All right, so in conclusion, LawLeave demonstrates superior zero-shot predictions for promoter indel effects across 3 complementary benchmarks. Autoregressive architecture enables complete promoter haplotype scoring, which is quite useful in these rare cases. This could be critical for clinical interpretation. And our clinical validation shows a 4-fold enrichment of known developmental disorder genes with this model. So next step would be to extend to, you know, other areas, larger promoters, etc. The world is your oyster. Apply to large biobank data sets to discover actual novel disease associations, guide target functional assays, and of course combine evolutionary evidence with you know, molecular evidence to enhance our predictions further. So with that, I'd like to especially thank Debbie Marks and the whole lab, and, you know, to thank Genomics England, who, you know, tolerated all of my airlock requests. There were a lot. And if you want to check out the code, it's kind of working. Maybe I need to spend a little more time with it, but I think everything should be up on GitHub and Hugging Face. I want to make sure that all the weights are like loading correctly. But if you want to play around with it or email me, be my guest. Thanks.
Great, thank you. Yeah.
Hi, wonderful talk. I had a question about the beginning. I think you had mentioned, correct me if I'm wrong, something about the directionality of these promoters, or the orientation of them, as also being something that can that you'd like to predict the effect of. And I was wondering if you had looked into how your model performs on that.
I believe, just to make sure I'm understanding, you're talking about the EQTLs. Or are you talking about the direction of the promoter, like
Yes.
in terms of strandedness?
Yes.
Oh, so the model does fine for either direction, because when I train it, I actually train on both directions.
Okay.
Yeah.
2 quick questions. One is, I was wondering if you explored the genetic capabilities of the model, like generating synthetic promoters. And the second one is, can you share more details about the model itself, like the differences compared to other models? Is it like a larger model trained on more species, or are there any technical tricks?
Yeah, so for the first question, I haven't really explored the generative capabilities. This is not something I've gotten around to. I was more focused on the prediction, but I think there's a clear direction for maybe the next student to explore the generative capabilities or to expand this modeling framework further. And the second question, so the model is not particularly unique, I would say. It's 200 million parameters, and there's we just use all the data from Xenomia, so it's just like 447 species. Yeah, and then I have a plot in the supplemental that kind of shows the drop-off of, you know, the number of promoters that are being kept across species. So like some promoters obviously aren't as conserved, and in my comparative genomics framework, they'll kind of like drop off if I'm not confident about them because I'm not relying on annotations. So I try to only keep things that, like when I was doing the, you know, sequence alignment, we're forming fairly large contiguous pieces. And if they got too sparse, I just like threw them out because I felt like I couldn't say anything. I wasn't sure. So there's definitely, I think there is probably some improvements in maybe leveraging annotations to build the training set like a bit bigger, build some confidence in there. But I was kind of, you know, just doing the most obvious bioinformatics I could do, I guess, at the time. Yeah, like I said, the model is a control transformer that I literally just took from, I think it came out in 2018 from like Salesforce, trained on Reddit data, and they just kind of like changed it a bit and added a few regularization tricks for dealing with like length and variance and things like this.
Okay, I didn't see any more questions, so maybe we go to the break. Thanks to all the speakers again for.
And well,
We'll reconvene at 5 to noon, so then we'll be only 5 minutes behind. So yeah, so whatever that is. 23-minute break, I guess.
Yeah.
Slow on YouTube.
Oh yeah.
You know how to.
I don't really know because it's going through this. Yeah, I don't know. They can turn up their volume on YouTube, I guess. I don't know. Maybe they're maxed out already. I was asking Nico Rabin because he was watching online. He said it was okay. But
Yeah.
thank you for running around. Yeah, I guess it's anywhere.
Maybe
It's
it's
good.
like even.
Maybe here is good. It seems like a good centralized place.
Everything depends on.
Well, the next talk is actually only, yes, I think you are actually okay. It's only one talk. It's just Alan's talk, and then it's the spotlights, and there's no questions in the spotlight. So it's like one talk, and then one oral, and then spotlights, and then lunch. So
Cool. Okay.
all right, I need to get a drink or something because I.
Yeah. Exactly.
Good.
Exactly.
Yeah, happy to.
Yeah. You're joining for dinner, right?
Yeah.
Okay, good. Yeah.
Yeah, he did like a hierarchical kind of like where all these like gene like the biological hierarchy. Yeah. I can ask this question but like it's not like it's the same rule as the thing. Also like similar thing. Yeah, why is, so these are like, all collective mailbox, so. For that is. Sure, like, I think it sort of, yeah, spun off. This is important, this is used in this, they don't know, like, right? I mean, yeah, but it's not like. What makes this like, I mean, it's not, it's really more hierarchy that exists in abilities, and it's just like, you know, well, like, most time, to symbolize the, you can, like, the environment, but like, it doesn't like me. It's, and that's kind of like, by design, and so the hell, it's just not your other ones. What functions use the, okay, and also like, because you're injecting all of like, this one sentence. But then, the network doesn't know that's a piece. Yeah. Right, so it's like,
I realized I forgot to have. Okay, so
what pathways are they like this? Like, let's say if you're doing the initial, you know, it's not, yeah.
yeah, that's something I think that happens.
Yeah. That doesn't really look about speaker. Yeah,
Oh, yeah, not that part. I'm not thinking about that yet.
Yeah.
Yeah, it's awesome. I guess not, ignorance, yeah, I'm applying, so isn't there? I guess. Yeah, not this enough, I think. And then, yeah, it's like most of the business.
Yes, that's one option. I think so. Also, like, my son was
I like how it's like well, there is, but like somebody's
I saw this live. I think we also technically Alright. You can take it private.
Oh, thanks. I know I owe you an email. It's one of those things at the back of my mind.
No problem, no problem. I know.
Yeah, it's a crazy summer. And then, I don't know, organizing this, it's a lot. Are you doing your laptop or you're doing? Yeah. Yeah, maybe you should just check. And are you in the Zoom? That's the other thing.
Join right now. Oh,
Yeah.
I don't have the Wi-Fi.
Oh, it should be NYGC. Yes.
Okay.
They made it easier now where it should just.
Nice.
If well, go to Chrome and see whatever your favorite browser is. Safari. Sometimes stuff works for me in Safari and not in Chrome. Yeah, United Wi-Fi now. I can only get on through Safari.
Okay.
It must be something's cached in Chrome. It's like
you're not connected.
okay, just try and go to anywhere, I guess.
Gmail.
Yeah.
Okay,
Okay.
let's try
No, okay.
Chrome. Okay, this is my games and such. Okay.
Yeah.
Gmail. No internet.
Freedom was a big Hollow Knight fan.
Okay. Yeah, that's right.
You know who else is? Do you know Bianca Dimitrescu?
No.
She's good.
That's
You
the
when you
only
get
game
on
I play, to be
Yeah,
honest.
she is awesome. I've played it. I guess maybe unjoin the Wi-Fi and join it again. I also have your laptop will be happy with. Okay, awesome. It's meant to bring up. There's no password or anything, but it's meant to bring up like a pop-up. And then,
Can I be joined? No network. Okay, maybe if I somehow forget it or something like this. Get this right here.
we can also we can also do it off mine, and if you.
That sucks.
Do you want to send me slides?
Yeah, sure.
Yeah.
I
sent a
That's
Dropbox link, or you can just email them to me also.
Yeah, let me just.
Oh, you can't wait. Oh, but you're not on the.
Well, I can just hotspot for now, right?
Yeah.
Send you something.
Yeah. I mean, I guess you could hotspot for the Zoom, but that's
Yeah.
depends on
Oh
how much
my
data you have.
God. Okay.
Don't worry, it's always.
Yeah. Da, da, da. Which one of these emails is the?
So I think if you click on this one, yeah, it should be, yeah, that Dropbox.
And Keynote's good.
Yeah, Keynote's good. Yeah.
Yes.
Yeah. Yeah. I've had people do the thing of converting my Keynote to Dropbox to PowerPoint without telling me and then destroying everything. Yeah.
Yeah, sure.
Talk was great, by the way. I have questions, but I didn't want to keep things going there.
Yes. Okay. Yeah.
You can also just email them to me, I guess.
Oh, yeah. Okay.
Oh, no, okay. That looks like it's going. So
4 minutes.
wait, really? Oh, because it's over your, it's on your phone. But you know, I have a USB key, dude. You know what? I actually bought it. I did actually mean to. I think I have a USB key. I think it's in my bag upstairs.
Oh, less than 1 minute left.
Okay.
Okay,
you
finish uploading.
You need transfer.
Yeah.
I'm just on hot spotting right now.
I have an external hard drive.
No, I think we got. I think we got it.
Okay.
I think we should do Jacob, right? Yeah. I mean,
that's the one. Lots of blue.
Should we just pull them from the spot?
Yeah, I guess it makes sense having to do the spot.
Okay,
I guess
sounds good. I'll let him know.
Yeah.
Now, if anything goes wrong, I'll be like, Dropbox.
Yeah.
You know.
So it's all its fault.
All
yeah,
the statistics are
exactly.
wrong.
Yeah. Oh, right. Yeah. It messed up the math.
Yeah.
Sure. Very convincing. How's everything been going?
Yeah. Pretty good. Yeah, I mean, half the apps this fall, so a little stressed out, but otherwise, and also, I cleared it. So,
Oh yeah, that's it. Yeah.
so it never ends,
Yeah.
How about you? Crazy something?
Yeah, I was stuck at. Well, I was meant to be in the UK for like 3 weeks. Ended up being there almost 5 weeks. I was renewing my passport, and it was. I don't know. I was originally going to do it from the US, and then I didn't want to be. My dad ended up in hospital briefly, and I didn't want to be like stuck without a passport and not being able to travel if it got worse. In the end, it wasn't that bad, but I was like, okay, I should just do it from the UK.
Yeah.
But then I was changing the return address. And they were like, His Majesty's Passport Office were like, no, you're not changing the return address. I was like, if you send the passport to America, I cannot get back in the country. So anyway, that became a whole thing.
Jesus.
But sorted now. So
Nice.
I have a shiny new passport. The frustrating thing partly was the US will let you in with just a green card, which I have, but no airline will let you on the plane.
Oh my
if you don't have a
God.
passport.
That's
so it's
so
like
annoying.
the US would let me in, but so.
Damn. Well, it's nice being Canadian,
That helps. Yeah. Actually, I was thinking if I was in Canada, I could have driven in.
Exactly.
So yeah.
There's back dark again.
Oh,
We just have to
wonderful. Yeah. Oh, they're on now.
I went back. Let's come on.
Okay. Thank you. Okay. Yeah, and that's good. Yeah, I don't know how to get rid of the little box in the right. So that is what it is.
Looks like it's actually zoomed in.
Oh, it's zoomed.
Maybe you zoomed in on the keyboard.
No, this is not zoomed in on here, right?
Why is this like?
Well, I'm trying to get rid of it as well, but yeah,
It's just this first slide, which is somehow really.
I think it's another slide too.
no, it's definitely no, it's got off the song. You're right.
Yeah.
Oh, weird. Look at this
I
thing.
think it's because you're sharing this window. Maybe stop share and try again.
Yeah, or share the whole screen. Maybe I've had that before. That sharing the whole desktop is actually more reliable. Yeah, nice. Okay.
Nice. Okay. Yeah.
I should know this. Amin or Amin?
Amin.
Amin, yeah. Okay, we'll get started again, everyone. Thank you for coming back. Roughly on time. So we have one oral from Alan Amin from NYU, which is going to be great. And then we'll have spotlight presentations. So if you're giving a spotlight, please sort of be ready to come up after Alan's talk. Oh, yes.
All right. Thanks for having me, guys. I'm Alan. I'm a faculty postdoc fellow at NYU, whatever that means. And I typically do projects around statistical foundations of discrete sequence data, DNA and proteins. So this is my first foray into disease data. So, well, keep that in mind. Okay, anyways, this is presented at ICML 2025, and we recognized at a workshop in iClear as well. So the task we're going to be looking at is a very classical GWAS setup. So we have a bunch of variants throughout our chromosome, and we're interested in predicting the effects of each of these variants on a particular phenotype, let's say a disease or some BMI or something like this. And the way we're going to do so, we would like some sort of information around each variant to predict its effect on phenotype, and we're going to do so using all sorts of functional annotations, right? So we have binding, conservation, other sorts of predictions, and we'd like to integrate all this sort of data using some sort of function to ultimately predict this effect to, with all sorts of downstream applications, right? And so this is, very much like the sorts of setups we've seen so far. Maybe where we'll diverge a little bit from Borzoi and Informer is how we actually get such a prior, where we get F theta. So you might think about F theta as, okay, we're going to take all these features, we're going to predict, expression or something like that, and then we're going to somehow combine all this expression data to make a prediction about phenotype. The paradigm I'm going to be looking at here, which has also been explored in papers like stratified LD score regression or something like that, is learning our prior using the disease data itself. So, let's say you have 22 autosomal chromosomes. What we'd like to do ideally is fit F theta on a subset of these chromosomes and then apply it to make our predictions about the effects of variants on this other held out chromosome. So this is the paradigm we would like to apply. We would like to learn F theta directly from this, let's say, phenotype data from like large scale you could biobank, let's say, this sort of stuff. And so the reason we would like to do this, of course, is that, this sort of data is exploding. There's a lot more, as people have said, right? And so what we would expect is, bigger GWAS data, and, we have all these transformer models now, and so maybe we can fit an F theta, which is huge, right? And like really expressive and all this sort of stuff. In reality, that has not happened, right? The F theta that people get from this sort of paradigm is at best linear. And the reason isn't because of data. It's not because of the models. It's because of something else. It's because of computational complexity and the algorithms that people are using to fit these data. So let me describe what the main algorithmic complexity is. Okay, let's say you have a bunch of variants here. And what you ultimately like to predict is the effects of each of these variants in a window of the genome. And we can represent them as bars as such, right? Now, what you observe is not the actual variant effects, right? What you observe is the association between each variant and a phenotype. This is what you get from UK Biobank. And this is confounded by or convolved with LD, right? So a variant can be strongly associated with a phenotype simply because it's associated with another variant which actually causes the phenotype, right? So what we have, the data we have to actually learn from is this observed association. And when we make predictions, what we would like is the blue bars to look like the gray bars, but we have to fit it to these black bars, right? Essentially. And just simply calculating the likelihood or accuracy of our predictions involves inverting this large LD matrix, and that's essentially going to naively be O of n cubed computational complexity, right? So this has been an algorithmic problem in fitting this sort of data so far. So people have all sorts of strategies, which I'll describe going forward. But essentially, the bottom line of our talk or this project is that we're able to get it down to roughly n squared. And that's going to allow us to actually start training very large F theta, which are transformers, 100 million parameter models. And that's going to actually result in better, more accurate predictions. Okay, so that's the little vignette. Okay, so what's the basic idea that allows us to build much more efficient algorithms for fitting these sorts of models? Okay, so here's the very classical GWAS setup. We have our vector of n phenotypes. N is the number of individuals we've measured. We have our n by n matrix of genotypes, right? And we have some noise, IID. Let's say it comes from a normal distribution, and this is ultimately the vector we'd like to predict, this beta. This is a vector of m effect sizes. M is each of those variants, and each beta m, right, comes from a normal distribution, 0. So the mean is 0, and the prediction of our model is going to take in a bunch of features around the variant m and output the standard deviation of the beta. Okay, so this sort of model, okay, and I should also say any fixed effects like top PCs, sex, age, stuff like this, I'm assuming have been projected out of Y and XT. So those are accounted for in that way. Okay, so why this sort of model? Well, this is can be arrived at from very classical mathematical genetics literature, like so-called conditional infinitesimal model. It can also be arrived at from like statistical foundations, sorts of assumptions about exchangeability of features. From a modeling perspective, this is really great because F theta outputs the standard deviation of the beta m's. It doesn't directly predict the effect sizes, so it sort of represents its uncertainty automatically, right? So compared with the sort of model Alexis talked about, fitting Borzoi or something like this, the predictions would be the mean rather than the standard deviation, right? So this is really nice because it automatically represents our uncertainty in an Bayesian framework, and so we're going to be, that's pretty great for fitting very large data sets with very high noise or very low signal to noise consistently, right? So this might be good for especially, fitting directly fitting phenotype data. Okay, so this is our setup, right? And so what we ultimately want to fit is this F theta, right? And so we're going to write down a marginal likelihood and try to optimize that. That's what we would like to do. Okay, before then, one thing I should say is that Y and X are not available because they're actually private. To many of you, you're like, well, I have the data on my, UK Biobank subscription. I'm poor, so I don't have that data. So instead, I had to use what's called public data. So the data that, and so let me just rephrase this problem to write it as a generative model or a statistical model of public data. So essentially, we're going to take this equation and just multiply both left and right hand side by X, the matrix X. Okay. So here's that exact same problem. On the left hand side, what we have is X times Y. That is the observed associations between each of the M variants and the phenotype Y. And vector of M variant associations. On the right-hand side, we have this X transpose. That's called the LD matrix, right? The empirical LD matrix, and that just measures the statistical association between the presence of individual variants. Okay, so now we have, and the nice thing about beta hat and R is that they're publicly available. You can just download them. Okay, so now we have an equation. Here we go. The explanation of GWAS data. We have beta hat comes from this sort of distribution. We get a likelihood, which is actually identical to the likelihood plus a constant of the Y model. Okay, so now we have all these terms over here are at the bottom here are things we observe. The capital F theta is just a diagonal matrix of our predictions. And now we just want to, let's say, maximize the marginal likelihood of this model. Okay. So there we go, diagonal matrix of predictions. Okay, so here's we can write the marginal likelihood out as such. Beta hat comes from a normal distribution as such, and we have A theta, which is our massive covariance matrix, right? M by M. So what we have to optimize is the log likelihood of our predictions, theta, which comes down to calculating the log determinant of A theta, and then, calculating this sort of expression, which involves inverting A theta. Now, calculating the log determinant of this M by M matrix and inverting it is going to be O of M cubed. Okay, so big problem, especially when we're, let's say, training by gradient descent, right? So every time you update your theta, you need to recalculate this whole thing again, right? Which is going to be extremely expensive, right? So, of course, we have GWAS models and people have been able to do this sort of inference before. But unfortunately, those sorts of previous methods are either toss a lot of data, they're not very data efficient, or they're not very compute efficient, or not compute efficient enough to fit very large high dimensional theta. So some strategies in the past have been to approximate R inverse using a low rank approximation. There's also a method of moments estimator called LD score regression. Those are bona fide methods you could use to fit these models, and I'll come back to them in the end, but they're very statistically inefficient, and there's a lot of literature about that. Another very popular method is variational inference. This is what Bolt LMM does, right? But unfortunately, those sorts of methods, they just manifest how statistically inefficient or computationally inefficient they might be. They fit theta using grid search, right? That's not going to work for a 100 million parameter model, right? So, in those models, they have 1 or 2 dimensions. And the advances in these sorts of models have been, moving beyond linear models, really sorts of, or applying very interesting prior information and priors, all this sort of stuff. We're interested in very high dimensional theta. Okay, so here's the basic idea of our rearrangement, it's actually very simple. We had a quite a complicated solution originally, and then after our paper was accepted, we found a much simpler one. So that's always nice. So here's our inverse, and it's a very similar rearrangement for the log determinant. So here's our inverse, and this is a sketch of the derivation. The real derivation involves careful handling of rank degeneracy of R. So let me just assume that R is full rank, and I'll some other stuff. So it looks a little bit more complicated in the paper, but here's the basic idea. Okay, so here's the matrix we want to invert. You apply Woodbury, you get this much more complicated looking expression. Okay, but you can simplify it as such. Okay, so you have this blue matrix, which can be written as B theta. Now, what's this thing on the left-hand side? This is if F were 0, that is, there's no genetic effect, that would be a theta inverse, right? So this is essentially assuming there's no genetic effect. Everything is just noise, right? Okay, so that has a nice interpretation, and also doesn't depend on theta. So we can actually just basically ignore it, right? And the other side is F theta is just diagonal, so pretty easy to deal with. All we need to do is invert B theta. And it turns out B theta is the identity plus what is typically a pretty small matrix. So what that means is that B theta is usually well conditioned. And that means that, yeah, this is the perturbation of A inverse theta from genetics, from genetic effects. And since B theta is well conditioned, right, its top eigenvalue divided by its smallest eigenvalue is roughly close, 10 or 100 rather than 1 billion, right? We can invert roughly in OM squared times using so-called iterative methods, right? So these are methods that just basically take, well conditionedness into computational efficiency. Okay, so that's basically the basic idea. Okay, so here's a comparison. So on CPU, we can apply iterative methods to A theta. It takes a very long time to invert it. You can see it takes over a minute. We can apply Nystrom preconditioning to improve the condition number of A theta. These are some off-the-shelf methods people use for Bayesian inference, large-scale Bayesian inference. Hardly an improvement. Cholesky, this is what people use in population genetics. Looks the same. Here's beta. Okay, look at that. Much better. And then moving to GPU, we get even more dramatic improvements, right? So the third bar is what people have been doing in the Nature Genetics papers. And then our thing is way on the right there, the tiny little bar over there. So, that's, I don't know, a modest 3 orders of magnitude improvement. Okay, anyways, so now we can actually start training our massive models using gradient descent on the marginal likelihood. So we get here's one of the models we trained, right? So DeepWAS is a pretty flexible nonlinear model. This is what we called our model, DeepWAS. So this is, I think, 140 million parameters. So here's a example of its predictions in a promoter region. Here's a few different features that we reasoned it was using to make these sorts of predictions. And you can see the predictions are pretty reasonable. So you can see that it predicts variance in regions that are highly conserved, as predicted by fastcons. This alignment-based model are predicted to be have a much higher effect on phenotype than regions that aren't conserved. You can see there's also some correlation with transcription factor binding and et cetera. Okay, so now, I'll zoom through the semi-synthetic data a little bit, but the idea here is we don't have on real data ground truth effect sizes, right? So what we did in the semi-synthetic data was just pretend, in a setting where we have the ground truth effect sizes, can we recover them using our more flexible model? So here's how we simulated the data. So we used real LD, but we simulated the effect sizes. And then we basically said, can we recover that? So constant means we tried to fit a complicated effect size model, a very nonlinear effect size model using a constant F theta. It did quite. Here's its error, using LD score regression or our, full likelihood model, constant, linear, roughly the same. When you use a transformer, a very flexible model, you're able to fit flexible feature data. Okay, effect size functions, let's say. And, using the likelihood was slightly more efficient than LD score regression. The difference between DeepWAS and LD score regression is much more dramatic when you actually apply it to real data. So so the y-axis is a little involved to explain, but essentially what we did is we trained on 3/4 of the chromosomes and then applied our predictions to the other 3/4 of the chromosomes. And the y-axis is just the percent likelihood increase in our, how well we explain the effect associations we see in these individuals. So constant is roughly, constant 2% means going from no genetic effects to having any sort of genetic effect increases the likelihood by 2% in BMI, 9% in height, very little in asthma. These numbers are, roughly correlate with the heritability of each of these traits. Okay, next we have linear. So we start incorporating functional annotations, and we see, DPOS does much, much better than LD score regression. Now, going from linear, which is what people have been doing in the past, to transformer, what we saw is that often LD score regression actually sometimes does worse, right? Or there's hardly any improvement. And that could be because of its low data efficiency, right? What we saw when we applied DPOS was that, you actually sometimes get as much of an improvement from linear to transformer as you did from constant to linear, right? So the improvement, I think of that as basically free, From constant to linear, you need to collect all sorts of functional annotation data. From linear to transformer, you just make your model size bigger, right? Okay, we did a bunch of ablations and stuff like that in the paper, but I'll end here with just future directions and acknowledgements. How far can we scale? We did some ablations showing that smaller models do worse. What other data can we incorporate? So obviously learning between disease sets might be interesting. What have our models learned? Digging into that might be interesting. And then also, incorporating this sort of, we talked, even Borzoi and Form can be used as pre-trained backbones for F theta, for example. So that might be an interesting way to, incorporate whole genome variant information into these models as well. Okay, this is my co-first author. If you think this is a weird picture, this is the most normal picture of this guy that exists. Okay, and then Andrew. Okay, thank you guys.
Testing. Hello, first of all, thank you for the awesome talk. I always enjoy geeking out about linear algebra. The question I have for you, I started looking at your GitHub repository whilst you were talking.
Yes.
And I noticed it's all in PyTorch. And given a strong emphasis at the start around, oh, look, you can make this really speedy. Is there a JAX-based or compiled language-based future to this, do you think?
Yeah, I think so. So what we used was a lot of the complicated linear algebra is a lot of the overhead is just in the kernels and such, and the particular libraries that PyTorch uses as the backend. JAX, I believe, uses pretty similar backends. In fact, it would be pretty simple to just extend to JAX because the linear algebra framework we were using was one from Andrew's lab called COLA, so compositional linear algebra, and that one's implemented in both Torch and JAX. I did in Torch because implementing, trying out different models is a little easier, but COLA is very easy to transfer between JAX and Torch. I don't think there's any free speed up just from moving to JAX, but yeah, that's my thinking. Yeah.
Okay, yeah, jumping on that point then, typically you probably would, specifically because it's an iterative method. These are the kinds of methods that PyTorch doesn't optimize very well, and actually there is meaningful overhead coming in from the Python interpreter.
Yeah, so yeah, that's a good point. Okay, so how much overhead is there potentially from the iteration? So how is COLA implemented? I believe, okay, you know what? I won't speculate about that very much, but I will say that most of the overhead, because in practice, the B is very well conditioned. The number of iterations we need is actually very small. It's typically 20 or something like that. Most of the overhead comes from the multiplication with the R inside the definition of B. So it's not so much, and that should be R, not R inverse. Sorry about that. It's from Dropbox. That's from so I wouldn't expect just a for loop of 20 to have that much of an overhead, but compared with the large multiplication with R, which is a very large matrix. But in general, yes, you could also compile the for loop in Torch as well. And I don't know whether they've done that in Cola yet, but Andres made Cola and he did it in both things, and he speculated there wouldn't be that much of an overhead from this particular thing in our setup. But in general, I think that's a clever point you're making. Yeah.
Okay, cool. Yeah, maybe this is a point we can debate in the break or something.
Yeah, absolutely. Yeah.
I did actually have one other question, which is a nice segue from what you've written here, which is where there are iterative methods for updating a matrix inverse it's already kind of fairly close. And given that this is in the context of stochastic gradient descent, where theta is gradually changing, could you try doing something similar here? Has that been?
Yes, excellent. That was the original setup, right? In fact, we used, like you basically calculate A theta every so often and use that as a preconditioner, right? So that's exactly what you're suggesting. This ended up being just much faster. Part of that is also because, okay, that I'll leave it at that, but we can talk more about more on that direction and why I speculate that could be the case.
Awesome. Thank you so much.
Thanks.
I think we should move
I
on.
think we should, yeah, I think we
Okay.
Yeah. Okay, so we have the spotlights up next. So if the people who are doing spotlights can try to be sort of available, I will pull up your slides. We're skipping Jacob for a reason we will announce later. So I think it's Evan is first up. Okay, this should all be sharing still. Good.
Just arrows will.
Yes, totally. I'm just trying to remember how you do slideshow. Okay, all yours.
Thank you. Okay. Oh wait, it's actually moving. Oh wait, is it on a are we?
Oh, interesting. Yeah. Huh.
Autoplay.
Yeah, I guess so, huh?
Makes it extra challenging.
Yeah. Yeah, it's just gonna go really fast. This is because preview wants to do
I can just go ahead and say that I'm a postdoc from Cold Spring Harbor. I'm with the Kinney and Koo lab. And okay, this work focuses on genomic deep learning models trained on sequence function data. And once trained, these models can take an unseen DNA sequence and return a predicted biochemical activity, often with striking accuracy. Despite their success, these models are notoriously difficult to interpret, earning the reputation of a black box, seen here in the first panel. Attribution methods address this by quantifying the contribution of individual nucleotides to a model's prediction, producing an attribution map, seen here in the second panel, that can be visualized as a sequence logo up top. In these maps, high-scoring nucleotides often assemble into recognizable motifs, aligning with known transcription factor binding sites, and interpreted as a picture of the regulatory elements that contribute to the predicted mechanism. However, attribution maps only show us a snapshot of the mechanism and do not forecast how mechanisms change in response to mutations. As seen over here, we can have a drastic change. Understanding these sequence mechanism relationships is essential to connect genotype and phenotype with direct implications for evolution and disease. And for this task, we developed SEAM, which has a weird highlighting. Okay, sorry. SEAM is a method that will help researchers better understand the regulatory mechanisms inferred by attribution maps, answering what regulatory elements are responsible for the activity at a genomic locus and where are these elements located. In the first picture, we developed a sequence entropy approach to pinpoint the exact position of all nucleotides within each motif. SEAM also helps explore the mechanistic potential of different genomic loci, answering which mutations preserve or disrupt regulatory elements involved in a mechanism, which mutations are neutral, and which create new regulatory elements in specific contexts. In the second picture, SEAM discovered a pairwise mutation in a cancer oncopromoter that radically altered the mechanism while reversing the direction of transcription, so finding mutations that matter. And SEAM also disentangles complex regulatory signals that we found are encoded in genomic DNNs, which we call foreground and background signals. In the third picture, SEAM isolates GC-rich background from the wild-type map, revealing a pristine set of foreground motifs. These findings position SEAM as a general framework for interpreting non-coding variants, distinguishing motif level from contextual influences, and guiding the mechanism-aware design of synthetic sequences. Thank you.
Yes.
Okay, thanks. Hi, I'm Suraj from Genentech, and I'll be presenting. We have a poster on NONA, which is a unifying framework for functional genomics. So we've seen models for functional genomics basically come in three main flavors: sequence function models like Borzoi, AlphaGenome, ChromeBPNet, variants of these that we call function to function that take in DNA sequence and some other tracks, and then predict some other tracks. So DNA plus accessibility to predict RNA sequence, RNA-seq, for example. And then also language models, so both autoregressive and masked language models. Examples are caduceus and EVO and nucleotide transformers. Key idea behind Nona is that it represents all of these data at base resolution in this big matrix that is multimodal in that it has different tracks and also DNA. And then you can mask arbitrary pixels in this matrix and fill those out based on what's observed. So what's not masked, basically. And what is nice about this is that it kind of unifies the three kinds of models I've shown you. They become instances of masks within Nona, right? So each of these different kinds of models I told you in the beginning end up just being different instantiations of Nona. But what's really nice is we can start going beyond these three classes of models, open up entirely novel modeling strategies that don't fall neatly into any one of these buckets, and open up some very new interesting applications. So in my poster, I'll focus on three of these novel modeling or like masking modes and their applications. The first is a context-aware model where we go beyond just DNA sequence. The second is a twist on language modeling where we condition language models on functional tracks. And what we also show is we can use these to design regulatory elements. And the third is what we call functional genotyping, where we go from functional tracks and we're able to genotype individuals without having access to any read level information. So check out poster 3, and also there's a poster 66 from my colleague Laura tomorrow. Thanks.
Hi, today we are going to talk about the deadliest infectious disease of all time known to mankind. In 2023, this disease alone has caused 1.25 million deaths and 10.8 million people fell ill. Today we call it tuberculosis, which is also a major contributor of antimicrobial resistance. The question we are asking today is, can foundational models predict antibiotic resistance from genomic data? So if we input DNA and protein data paired with phenotype data for candidate drugs and ask models like DNABART2 or ESM family models, if they can predict with decision and certainty, like a certain antibiotic will work for this strain of disease or not. And if we take TB antibiotic resistance as a test case, can a new benchmark dataset test functional models with this task? So I present our poster, Big TB, a benchmark dataset for genomic resistance, prediction, and interpretability in Mycobacterium tuberculosis. I am Mahbooba. This is my co-author Saisrada here with me. So we offer a proprietary data pipeline for producing DNA and protein from VCF files that's paired with phenotype data from isolates and also has resistance confidence levels at mutation level from WHO. And finally, this dataset is benchmarked on two tasks. Task 1 is phenotype prediction, and task 2 is causal variant discovery, which is a new introduction in this type of models. Our primary investigation finds that for a candidate drug like isoniazid, for both DNA and protein data, simple models as velocity regression is outperforming DNABART and ESM family models. So we have concluded that functional models, they are powerful, but not yet superior to simple models for antibiotic resistance. We have seen the similar trend in the causal variant discovery task too. Come see our poster at Spotlight 4, and thank you for listening to us.
So highly multiplex imaging technologies like imaging mass cytometry or IMC enable us to quantify over 40 proteins by retaining the spatial locations. Traditional IMC images are captured at a resolution of 1 micron, where we're able to capture single cells, but we only capture a very small section of the whole tissue. So a recent development in the space has been the acquisition of whole slide IMC images. These images at a resolution of 5 micron are able to capture the full heterogeneity we see in the whole slide. But in comparison to the 1 micron, a thing that you can see right away is that while we capture a much larger tissue context, we lose that single cell resolution we had previously, meaning that the standard analysis tools available can't be used because they all relied on cell segmentation as the first step. So the question here becomes, how do we analyze these images when we can't segment cells? So our approach is that why not take advantage of seeing these much larger tissue structures, such as ducts, and try to segment these instead. For this, we look towards foundational models in computer vision, such as the Segment Anything model, or SAM, which has shown remarkable performance over a variety of segmentation tasks. A challenge here, though, is that it was trained on natural images, which only have 3 channels, whereas IMC has over 40 channels. So what we propose to do is that we learn a channel number agnostic projection matrix that takes in an IMC image and gives a 3-channel projection that then serves as the input to SAM. We call this a low dimensional fine tuning of the model, and we train this end to end by comparing SAM predictions to ground truth annotations and backpropagating through the whole thing. So if you're interested in how we actually train this model, whether or not it actually works, spoiler, it does, and any insights that we've gained by applying SAM on a novel 5-micron prostate cancer data set, come check out my post number 5 during the poster session. Thank you so much, everyone.
Sorry. No. Okay, hello, my name is Pavel and I'm postdoc at Joe's lab at UT Southwestern, and today I present our take how we can improve genomic data with sequence-based models. Of course, for genomic data, we would like to have single cell specificity and base pair resolution. I believe half our problems would be solved if we had this data regularly available. Here's an example of an MSeq2. So we have single cell data, and if we aggregate 8,000 cells, we have these nice profiles. And of course, we would like to have single cell on this level. And limitation is technological costs, availability of biological material, and many others. And people usually try to develop a computational methods to improve resolution, and we decided to develop another method for this too. So, and we base it on recent success of sequence to function models. So we know that they generalize well. So, and we, in addition to genomic sequence, we provide a low coverage experiment, and we developed a new training technique to prevent model to just copy the data. And the key features of our SURF model is it doesn't require high coverage experiment. It generates high resolution, low bias, low variance estimates. It's widely applicable to different genomic assays. And we developed some statistical guarantees as confidence intervals and likelihood to measure performance of this model. And just to highlight the results, so we achieved for different coverage level, for example, for DNA-seq is 100x improvement in coverage level. And if we focus on single cell, we see that we can boost single cell tracks to essentially bulk level. And if you would like to learn more details, come and check my poster.
Thanks. Hi, I'm Simon, and I'll be presenting a preview into our work with distilling RNA splicing logic from deep learning models. To start with some background, alternative splicing is a process that enables a single gene to produce multiple distinct RNA isoforms. One of the major outcomes is whether a particular exon will be spliced in or out of a final transcript. So deep learning models like SpliceAI, Pangolin, AlphaGenome, they can all predict splicing with pretty high accuracy now. The problem is that they're mostly black box models. So as we've heard today, they learn really well. We don't really understand what exactly they learn, which is a problem when trying to study biology. And so our approach was we developed a framework using model distillation to try to extract an interpretable splicing logic. So this distillation consists of training an interpretable student model from SpliceAI or Pangolin to try to extract something that we can then interpret. And so first, we generated 1 million synthetic sequences with a randomized middle exon and asked SpliceAI and Pangolin the likelihood of that exon to be included or not. And then we took those generated scores and sequences and trained an interpretable model, which consists of using short sequence features with additive contributions to make a prediction. And we found that our interpretable models performed very accurately at reproducing these exon inclusion scores. So then when we actually interpreted these interpretable models, we found that they identified many known splicing motifs that correspond with exon inclusion as well as skipping. Surprisingly, we found that they also learned motifs that don't appear to be directly related to splicing, such as CpG dinucleotides and stop codons. Instead, it seems that deep learning models are learning these because of co-occurring biochemical processes in the genome, such as methylation or translation. So in summary, we developed this framework that allows us to make highly accurate and interpretable splicing models. If you'd like to learn more, please come check out our poster. Thank you.
I think that's it, right? Yes. Okay, so thanks everyone. We'll be back at, I think we can get back on time actually, so restarting at 1:30. And yeah, lunch, hopefully pizza's here. Haven't had a confirmation, but yes, it is. Okay, awesome. You get 2 slices to start with, so watch your figures, yeah. Unless you want other people to go hungry, so.
How you doing, Mark?
Frustrated about this. You know, the worst thing is they checked it yesterday and it worked fine. Good point. Yeah, it's frustrating that my admin and like IT people checked it.
And then the bulb.
I guess I don't know. I mean, it's quite, maybe the bulb. Yeah, I guess so. I taught. I was teaching every other lecture of my Columbia class in here when I was teaching.
Okay, I'm very short time. Yes. Now, we got especially if you want to include something like that. Maybe in person you can. Have that kind of direction openness, you know? So, you guys see that, like, this is right now. The problem is okay, I'm saying it's not like this kind of thing.
Today I told you I said it there.
That's right, so I mean, those it's like, exactly, it's the same thing, that's what it is. Exactly, right. That's right, I mean, maybe I should add that in, aspects of some. Seeing how many. Yeah, so I mean, yes, that's right, that's right. So, yeah, that's right. So each variable. My sister, I really just picked them like I was making and so I was asked like aggressive, only the highest quality, these roots were like some of these roots, right? I found like, you know, this, a bunch of other predictions, and so I just kind of like made some so that really they actually like Chinese. So I'll show you an example from the one of the fonts like predictions, and below that was the features. And so they emails, I think it was like 160 by 512 right? And you know, you'll be taking that big matrix and just output a single number there. And it should be the log of lyric.
Next week, but you know.
I mean, the way that you said that problem, you know, when you say, we all have problems of the work. Yeah, you have
like the real one, and we say, so, okay, so we have this one data set, which is what they can about it, right? And, you know, I ultimately how this product is to produce is to like fine tune it on itself or like have like a train on itself and apply it on that process. This is how, for example, like the hunter parameters element train, right? So like this was actually like all this data set you can use it. And made this prediction on that speed. So I agree there's like some sort of like illegal will improve learning. If you can actually work, I really don't know what I really don't know what I would say this. I know yeah, obviously, like you're not going to have to with this. It's to basically try and do a good flag. So basically, so one way to think about it is like that's also right? And that one line is doing like that's how that also is going to be also set and you know, like we have nations, we have posts, something like that. And you know, the model is more than 89, which is not going to have right? So this is what this column is looking at. And we just move private and ever stuff. I don't know much about. Yes, yes, that, we have those words we use have a big card. Yeah, it is a very big card. Which includes all the and what's the application? I don't know. What do you think about it? No,
Very cool. I didn't want so much. I do want. They even have to. Okay. Wow. Great. Okay. I'll be there in a minute.
Are you one of the organizers?
Yeah.
Nice to meet you. So how far behind are things running? Like when is the post session supposed to end? Is it still 1:30 or has it been pushed back?
The post session is not right now. The post session is afternoon.
Those at lunch and posters, 12:30. Okay, so I guess maybe we would just look at the posters now, but that's the actual presentation.
Yeah.
And so, but when are we meeting? Is it still 1:30 or is it like 1:50? What will be the
That's why I'm here because I think it's longer.
Okay, yeah. I just know things are running a little behind.
Yes,
But okay.
I don't think most people just gone through this. Okay, thanks.
It was supposed to be a big company on, I'm almost like. Yeah.
Um. Long-term scientist, of course.
Okay, so I uploaded my slides on the Dropbox that David sent around.
Okay.
Really?
Oh, do you want, I don't know, do you want presenter notes at all or anything? You don't need it, okay. Yeah.
Yeah, we met.
Yeah, we met, I think we met like a few times, but the most recent one was I did a job.
Oh, yeah, I know. You know what? I actually wasn't there that week, but yeah.
Yeah. You weren't there, but we did those.
Oh, yeah, the three training. Yeah. I remember that. Yeah, that was fun. No, fantastic.
Oh, was it?
Don't, there's
Yeah.
nothing to say. We actually failed.
Yeah,
Completely.
I heard.
There's a mismatch between.
Yeah.
Don't know, like on the thing, did they try cycling at points, you know, the main projectile?
Oh, no, I haven't heard anything about that.
Oh, thank God.
I think
There's a downside, right? Turning it off and on again, because there's.
No.
Right?
Yeah, we should try that.
Yeah.
That's in the back. I'm one of the mic runners. Yeah, I just tried turning it on and back on, make sure it works. Which side do you want to cover? Okay. What? I'm not introducing you. I'm the chair. But I think that is it Peter? Have you seen it? I'm trying to figure out. Hey, how's it going? Everyone doesn't have to swim.
Yeah, awesome. Yeah, that's awesome. Yeah, exactly.
Seems like it's 1 in 3 roughly every time you turn it on. Maybe it becomes
Great.
they tested it yesterday and it worked.
Of course.
I thought it was going to be developers out, so it's not all this stuff. I mean, they bought this stuff in like 2009. They
God.
set up the genome center,
Yeah.
so it's been sitting. It's been
Million
used for like
years
15
ago. Yeah,
years. Yeah, is it on in that?
It's not.
Like
2017.
Looks like something. Oh, that was totally worth it.
Yeah,
That was kind of ridiculous. The first half. Someone was supposed to do that during the break, but they did. I guess they didn't.
Yeah, no.
Wait, so you still have a footprint here, which or
I guess that's a fine, right?
yeah.
Yeah.
It's good. Yeah, I ended up deciding to stay at hospital, but then
Yeah, I bet.
it's like mids and like yeah, like validate our stuff or generate data. Yeah, and not rely on collaborative.
Right, exactly. Yeah. Story of my life, right?
Yeah.
Yeah, actually, I have a quick collaborator that has.
I'm gonna make sure.
This week, right?
Oh, yeah.
Does everybody have their their talks loaded, or we got to deal with?
I'll do it.
Okay.
It's a mix. Some of them uploaded them, some of them have their laptops, but they're like panelists on here. So
They're what, sorry?
they're panelists on the webinar. So then they
It's just
yeah,
so
great.
like
Yeah, I
stream.
know. I think we want unlimited. Yeah. Hey.
Sarah.
Oh, yeah.
Okay.
Hey, how are you? Good luck.
All right, we're gonna start the afternoon session now. So if everyone can tone it down a little bit, that'd be great. All right. Okay, so I'm very excited to introduce our next speaker, Barbara Engelhardt. She's a senior investigator at the Gladstone Institute and professor at Stanford University in the Department of Biomedical Data Science since 2022. Before that, she received her PhD from UC Berkeley, advised by Michael Jordan. She was a postdoctoral fellow with Matthew Stevens at the University of Chicago. She was an assistant professor at Duke from 2011 to 2014, and then moved over to Princeton, where she became a full professor. She has a diverse work history, from Jet Propulsion Laboratory, Google Research, 23andMe, and Genomics PLC. She has also received numerous awards, including the Sloan Fellowship, NSF Career Award, and the Overton Prize. Her research is focused on developing and applying models for structured biomedical data that capture patterns in the data, predict results of interventions to the system, assist with decision-making support, and prioritize experiments for design and engineering of biological systems.
Thanks for that really nice introduction, Peter. I've never diagnosed anyone with appendicitis, so I feel kind of bad about that. Okay, so today we're going to talk about some work that started 4 years ago when I moved over to Gladstone and Stanford. And so it's a little bit different. Oh, this isn't moving. Oh, here we go. So actually, we're going to start back when I was at Princeton. I had a collaboration with Jared Tocher, who was working on these types of data. So this is a picture of a fluorescent slide with canine kidney cells on it. And at the beginning of the movie, there's a wound being created at this spot over here. And you can see the cells of this in this slide basically coming over to try and heal that wound. And what you're seeing with the colors is blue is going to be low ERK signaling. ERK is a signaling kinase. Green is high ERK signaling, and then red is just the background of the slide. So you can stare at this 100 times, and I've probably stared at it 1,000,000 times at this point, but there's a lot going on here, okay? So first of all, we don't see very much ERK signaling going on at the wound frontier. We see a lot of really nice patterns of ERK signaling within the frontier, and we'd like to understand more about this. So just to sort of fast forward, after trying 6 other models, we ended up using what's called a Hawkes process to deconvolve 3 different drivers of the signaling. I can't tell if I'm talking too loud. It sort of sounds loud to me. So the 3 different drivers of signaling here are going to be a spontaneous pulsing rate of ERK, then the self-excitation. So if you, a single cell, fire ERK, what's the probability that you fire again? Within some time period, and then cell-cell coupling, where if you're a cell and you signal ERK, what's the probability that your neighboring cell is going to fire ERK as well? So this is sort of a visual of what a Hawkes process looks like, so you have an idea. It's a spatial temporal point process, but the idea is if this is cells 1, 2, and 3, if we see a pulse at cell 1, that increases the probability because of self-excitation that there's going to be another pulse at cell 1, but it also increases the probability that there's going to be a pulse in cell 2. And you can see as these pulses actually accumulate what the probability is that there's going to be a pulse for each of these 3 cells, and it depends on the neighbors there. So we built actually quite a simple model at the end of the day where the rate of pulsing has to do with the spontaneous pulsing rate mu. And then we have the self-excitation rate when J equals K here, when it's the same cell. And then we have the cell-cell coupling rate when J does not equal K here as well. And we can just really easily fit this model to the entire data set that we have and get the answer. So why was this fun to do? Well, no one had really played around with this data other than the people who initiated it, which are Aoki et al. From 2013. And they did the same thing that we did, to be totally honest, except they did a manual annotation of 5 cells. And they tried to pull out these numbers from 5 distinct cells here. So we were assuming that when we looked at all 300 cells, we would do a better job. And the short answer is we did. So in particular, we actually looked at 3 of these different slides. One of them was the control, the DMSO. The other 2 were treated with chemicals that essentially inhibit cell signaling. So we knew that for the cell signaling parameter, the rate parameter, that those 2 treated slides were going to go down in terms of the rate of signaling. We also saw, as you saw in the original video, that depending on where you were relative to the wound, and this is sort of flipped over, but here the wound in both slides or in both pictures is at the dotted line, and internal is the opposite direction. You see that there's basically no cell signaling right at the wound position, and as we go in, it goes up, and then it goes back down to some base level. You see also with the 2 drugs that are put on us that they both inhibit cell signaling, although actually back beyond the wound frontier, they don't actually inhibit cell signaling too much. What we found that hadn't been found before with this model is actually the base rate of signaling for ERK also decreases with these 2 drugs. So that was kind of exciting. So fast forward to me showing up at Gladstone, and I had lunch with Alex Marzen, and he told me about this experiment. Alex Marzen style CAR T engineering goes approximately like this. So his goal is to design immune cells, in this case T cells, that are able to kill cancers, cancer cells specifically. And so his sort of repeatable experiment is that he takes a bunch of T cells. The first thing he does is put a TCR virus on them that essentially allows them to have these cell membrane grippy sticks that allow them to grip specifically onto cancer cells and also be activated by those cancer cells. More than that, he actually does some kind of CRISPR-Cas9 knockout. So these are modified T cells. They're modified in 2 ways. They not only have the TCR, but they also have a modification and knockdown. And then what he does is repetitively stimulates them 6 times and then puts them in this co-culture. And I'll show you what that looks like in a second. And then over 48 hours, they take pictures of this co-culture at high resolution. Actually, it looks something like this, and I'll go into a lot of detail on this next. And we get to see what happens. So here is an example of one of them. So what you're looking at here, the bigger, longer cells are going to be cancer cells. The smaller, faster cells, the circular cells, are T cells. That first part that you saw with the small cells going up and around was actually just a pipetting artifact. But you can see a lot of stuff going on here. There's a lot of complex relationships. You get to see the T cells sort of as groups attacking these cancer cells. You get to see the accumulation of these clusters of cells. And it's also hard to see everything with so much going on. There's approximately 300 cells of each. There's also, as I'll show you, duplication and splitting and dying as well. So these are really complex experiments, and I'll say they don't always look the same. I showed you one where it looked like the T cells were doing a good job of killing, but if you just look at the black and white, this is from the fluorescence image, you can see that at hour 132, it looks very, very different. So here the black and white, the white signifies the cancer nuclei. So you can see again at hour 132, either you have like this really nice even spread of cancer cells, or they're entirely clustered around the corners. And we can talk about why that is in a second. So here's the weird part, and this is why I went over to Alex's lab after this lunch and with a hard drive and got a whole bunch of these videos. So many labs have InQSight machines. I'll say there's 8 within a spitting distance of my lab at Gladstone. So these are common, ubiquitous, cheap experiments to do. But for every paper that actually uses these data, they often just reduce them to this graph. So what this graph is over the time on the x-axis, you can see the growth of the cancer cells on the y-axis. This is basically just how much coverage with the fluorescent marker of the cancer nuclei are there. And you can see again that when you have a no knockout or no T cells, the cancer cells proliferate. When you have this control set of T cells, they still proliferate, but not as well. But when you have this beneficial knockout in the T cells, they actually control the rate of growth of cancer cells really nicely. Okay, so here's another example. Again, I can pull out a lot of cancer immunology papers where all of these beautiful videos are basically compressed down to something that looks like this, which is kind of a bummer. And we noticed by looking at the videos a lot that these cancer cell growth by time figures missed a lot of signal. There's a lot of interesting stuff going on in these videos that ended up on the cutting room floor, as they say. So we'd like to understand more about what that is. So again, we spent a lot of time just looking at the videos to start with. One of the cool behaviors that we see is actually there's this really interesting sort of pack hunting kind of philosophy that goes on among beneficial T cells, right? Where they together sort of gang up on one or more cancer cells jointly, sort of signaling to each other somehow. We also see a lot of cancer cell proliferation. We can see this in T cells too, but it's a little harder with these frame rates. So you're going to see this cell ball up and then split, and then actually this one over here balls up and splits too. You can see that this is even happening when there's a T cell hanging off of it. So even when there's a T cell that's trying to kill it, these cells can be prolific, which is kind of a bummer. We also see cancer cell death, but I'll tell you the sort of sad truth, which is actually the way that T cells tend to control cancer cells is not by killing them, but by preventing them from splitting. So it took us a really long time to find this good example of a cancer cell death, but in it, so you can sort of see the cell falling apart. And really quickly, just to orient you also, these cancer cells, when they're long and skinny, that's their sort of relaxed state. That's when they're essentially lying on the floor of this well. So they look all stretched out. When they ball up, they're either doing that to try and move and evade the T cells, or they're going to split, or they're going to die. So if they're round, that's a bad thing. I mean, it's a good thing in that they're almost dead or something. Okay. Why is this not moving forward? Okay. So again, 4 years ago, we sort of wrote down a whole bunch of things that we wanted to do. And we've done the top 3, and then I'm going to sort of stop but before we get to the 4th. So the first one is just building these time series models that essentially track the morphology, the motility, and the interactions between all of these cells. The second thing we really wanted to do was understand what these clusters was happening, the blossoming that we see of groups of cells. It's really hard to study that when we just do segmentation immediately. So we wanted to figure out a different way to do that. We also wanted to just characterize patterns. Can we use deep learning to actually predict what type of knockout we're talking about, which cells we're looking at for the T cells? We wanted to go one step further and do imitation learning, which basically is our machine learning methods that allow us to say where the T cells are in the next frame to try and figure out what's motivating them to move, I guess. And this is where we got stuck, but I'll tell you how we're getting over that. And we thought of other stuff too, but I'm just going to skip over that for now. So we developed something that we're calling the accident workflow. Accident is actually the name of the horse in one of the first moving pictures. And ironically or not, this movie was actually filmed at Stanford, which is kind of awesome. So not surprisingly, the first two things we need to do are segment and track every single frame in these videos. So as I sort of mentioned before, and I'll just go into details here, we image these TCR T cells. They're co-cultured with these A375 melanoma tumor cells after repetitive stimulation, so 6 times repetitive stimulation under 3 different conditions. So our safe harbor knockout, where essentially it's basically just a TCR T cell, so there's no beneficial mutation. And then we have RASA2 knockout and COL5 knockout, both of which were developed with Julia Carnevale and Alex Marzin's group, and both of them do slightly different things, and we're going to get to that in a little bit. In these data that I'm going to show you, they were imaged every 4 minutes at 10x magnification for 72 hours. The imaging actually includes 3 different channels. So we have bright field, which is just the picture, RFP, which marks cancer cell nuclei, and GFP, which marks T cells. But in what follows, I'm basically going to ignore the GFP channel entirely. Okay, so the first thing we did was collaborate with Dave Van Valen at Caltech, and he has a beautiful method that allows us to do segmentation and tracking. We'll talk about sort of the limitations in a little bit, but first you can see that he uses also the bright field and RFP overlaid on it, and using his deep cell method, we can really nicely grab the cancer cells. We actually, on top of that, used segment anything, as discussed earlier, to grab all the other cells, the detritus often, and then the combined segmentation worked really nicely to grab both T cells and cancer cells, pretty inclusive. So then the tracking basically used these masks to try and develop tracks. Notice what's going on here, that it's not perfect, and we'll talk about that moving forward. But I'll also note, just because this is a big issue, is that Caliban, which is his segmentation and tracking method, was actually trained on macrophage InQSite data. So it's just a well of macrophages, which are an immune cell that are basically the polar opposite of T cells, right? They're sort of like Jabba the Hutt. They kind of ooze, and they're big and not really round, and I don't know. Anyway, it was a very different situation than the one we're looking at. Okay, so now we can see what's going on after we've done segmentation and tracking with all of our videos. So here's an example where we just count the number of cancer cells. And here you can see that for every single experiment condition, the safe harbor plus RASA2 and COL5 knockout kind of go up to start with, but then they go down for both of the beneficial knockouts, whereas for the safe harbor, it kind of goes back up again. We can also do this with T cells, which we couldn't really do before. And this is exactly what we were hoping for, that for the safe harbor knockout, there's basically no proliferation of the T cells from the start, whereas for both RASA2 and COL5, we see that they proliferate a lot more than they need to. And this is a function of basically the activation from the cancer cells that they do a better job of. One thing you should note now, just to foreshadow what comes later, is that COL5 proliferates a little bit faster than RASA2. And we know this because the COL5 knockout is a gene that essentially inhibits proliferation. So we are fully aware that these genes these T cells were supposed to proliferate a lot more. Okay, so we can do a lot of things now, again, with our segmented and tracked data. So one thing we wanted to do was understand when cancer cells divided. And so as you saw before, when you have a T cell hanging off of you, you can still divide. And in fact, what we did was actually computed the change in division in the proliferation of a cancer cell per every single additional T cell that was attached to you at a specific time. So in safe harbor, again, they don't really inhibit as well, whereas for both beneficial knockouts, RASA2 and COL5, they both inhibit, for each additional T cell, they inhibit a proliferation of that cancer cell a lot more. And this again goes to sort of the story about how T cells don't really kill cancer cells sometimes they just prevent them from splitting. Another thing that is super cool about this that I don't think will come as a surprise to anyone is that cell morphology tells you a lot about cell state. So there are a lot of really cool things you can see just by staring at these bright field images. So for example, active T cells are a lot larger than non-active T cells. This is probably not surprising to immunologists because once a T cell is activated, there's a huge burst of transcriptional activity, but you can actually visibly see the cell get bigger, and I'll show you some data on that. Active T cells are also less circular in shape. I assume that's because they're growing really fast. Cancer cells aggregate when they're exposed to T cells, which we were excited to see. It looks like they're trying to evade attack. Cancer cells elongate when they're stable, which just means they're adhering to the bottom of the well, like I said. And then cancer cells become round when they're either proliferating or dying. So you can't really tell by the roundness of a cancer cell what's going on. So the first thing that I thought was really cool, just because there's so much signal here, is that the T cell size clearly shows activation after it interacts with a cancer cell. So as I said, one of the purposes of having a TCR, a T cell receptor on top of these T cells, is that now it selectively adheres to all these cancer cells. And when a TCR, when a cancer cell and a T cell are together, the purpose of that is really to activate these immune cells. So you can see in this really simple picture here that you have the T cell mask in green, you have the cancer cell mask in red, and as the cancer cell separates from the T cell, you can see the T cell is a lot bigger. The mask just has more pixels in it. So we did this across all cells and found the following, that if you look at the minutes relative to the interaction of the two cells, which is at this gray dotted line, before T cells were, this size going down slightly, and then as they interacted, you can see this really big burst for all three conditions of the size of the T cell in square microns and pixels. We also see that cancer cells shrink after they have this T cell connection. So with the same semantics as the one before, we can see that this dotted line is when they actually interact. This is the cancer cell in square microns, cancer area. And you can see that safe harbor starts out higher, but they all drop to about the same size after these interactions. We are assuming that they start out higher in the safe harbor in the controls because there are just a lot fewer T cells running around attached to cancer cells. So they start out basically healthier. But then after these interactions, no matter what the condition, they all go down to a pretty low square area, which is kind of cool. Another thing that we see is that cancer cells actually slow down. So we can see that they move around a lot before the interaction. And again, this dotted line is the actual interaction. And then their speed of the tracks that we have actually decreases after those interactions. We used a really simple spatial Markov model to try and understand, for example, the difference between T cell recruitment and T cell proliferation. So the specific thing we're trying to deconvolve here is if I have a cancer cell and one T cell in a small window, it could go to two possible positions next, or it could go to the same model next, which is a single cancer cell and two T cells in two different ways. Number one, it could recruit another T cell from somewhere else, or number two, it could split. I think I did that exactly the opposite. Yeah, it could split here. And we wanted to figure out what was going on in both cases. So, this is very light, but you can see that the safe harbor has very low recruitment, but it proliferates a bunch. Whereas RASA2 and CUL5 basically do twice the amount of recruitment, which is quite impressive. And CUL5 also proliferates more than RASA2. So there is a lot of different things going on here to be able to get T cells to help out with the cancer killing. Another question that we had was, and this is actually very much directed by the goals of these cancer immunologists in terms of making effective CAR T and TCR T cell therapies, is how long do these T cells stay attached to the cancer cells? So they either call this attachment time or dwell time, where again, you have a T cell coming up to a cancer cell, sticking with it, and then separating. And we really want to know how long they stay in this together state. So we actually used our Markov model to answer that question. We also just built a really simple negative binomial detachment model, and they found basically parallel things. So the mean interaction time for the safe harbor was sort of around 15 minutes, whereas for RASA2 and CUL5, it was more like 18, 19 minutes, which was kind of exciting. So we do see that they're attached longer. And actually, the important thing to note here is that this is where the RASA2 knockout works better, basically, than COL5. So maybe it doesn't proliferate as much as the COL5 knockout does, but it attaches better, which is cool. So to summarize all of this, the first part of the talk, live cell imaging, we're really trying to understand the cellular behavior. So we summarized all of these data by saying, compared to the control T cells, the TCR T cells with a RASA2 knockout have this longer dwell time, this longer interaction time, and they seem to cripple cancer cells more effectively just because they interact with them longer. In contrast, we see that the COL5 knockout TCR T cells proliferated more frequently. So they basically just swamp the space with additional T cells. And that's how they more effectively fight the cancer cells. So we actually have the tools to segment, track, and analyze all of these cells on a GitHub repository. One thing that I'm extremely proud of is that 4 years ago when we were looking online, and actually you can do this now, none of these videos are available online, even though every lab has stacks of hard drives with these videos on them because they're so easy to generate. None of these are online. So we actually called up the people at Bioimage Archive and asked them nicely if they would put them up, and they said yes. And we actually have all of the data that we used for this experiment, plus another one that we just put out on bioRxiv in June. We have all of that up on Bioimage Archive. So highly recommend going there, looking at these data. Also highly recommend encouraging other labs to put up their IncuSite data or live cell imaging data there. And this is our original preprint. Okay, so this brings us to the beginning of the summer. So here's the video that I showed you before. This is the segmented and tracked image. And I'll tell you what happened. So we had a summer intern who was going to do the imitation learning for us. The idea is, given the previous T-S frames, predict where each T cell is at frame T. So the idea behind this is not particularly exciting. We wanted to figure out what if we could predict where T cells go. If we can predict where they go, then maybe we'll understand why they go there. Do they see a T cell? Do they see a cancer cell? Why do they switch directions? We're just going to go in and do some explainable AI on the models that we built. So we messed around with this for probably way too long, did a lot of, smart things that people do when they're trying to predict things using machine learning and images, and basically totally failed. And we specifically failed in the sense that if you think about predicting the next position of one of these green labeled cells, these T cells here, actually none of the models that we built could predict better than predicting it was in the exact same place as it was last time. Okay, so audience participation moment. Can you tell me why? Just looking at this video. Yeah. Yep, they vanished, exactly. Yep, the tracks are short, basically. Yeah. Anything else? Well, it's bouncing around, right? The frames aren't even registered. So I think we need to start something else. So we actually went on 2 tangents based on this. Number one, what can we do without segmentation and tracking, right? Can we actually think about these models without worrying about how good the segmentation and tracking was? And number two, what happens if we actually have reliable segmentation and tracking? What can we do? So I'll really quickly tell you about the 2 methods. I have no idea how much time I have left. I'll really quickly tell you about the 2 methods that we developed for unsegmented and untracked cells. So the first one, which we put out as a bioRxiv paper in June, is basically using, so we tried doing this with a lot of deep learning models, and actually at the end of the day, these methods from the 90s, the shift invariant feature transform image filters ended up doing a really good job of getting us the answers that we were hoping to see. So basically, we put these SIFT image filters on every single one of our frames, and then looked to see if we could sort of cluster and figure out what each of them was marking. And it's kind of hard to see here because I have a lot of things going on, but basically, if you look at some of these features, you can see them grabbing T cells in spaces where there are healthy T cells in the space where there's unhealthy T cells, healthy cancer cell clusters, unhealthy cancer cell clusters, and individual cancer cells. We also got a couple clusters that were basically just the wall. So that was kind of actually good because we had this control and we could really easily see what was going on there. But yeah, we also found very interpretable clusters using these SIFT transforms after doing k-means. Okay, so really briefly, again, I'm not going to go into a lot of the results from this, but if we look at these healthy cancer cell aggregates across time, you can see sort of what you'd expect, I think, where if you have a lot of, well, it depends. So these different colors represent, different E to T ratios, which is effector cells to target cells. So the higher ones are the ones where we have fewer T cells involved. So if we look at, for example, the T cells that are effective, more T cells, we see that these unhealthy image feature, or these healthy cancer cells stay low. And if we see where we have very few T cells, we see that they go way up and then back down again. For the unhealthy ones, again, not surprisingly, we see that they go up pretty fast, even when there's not very many T cells, and they peak later, but they don't really come down. And then for lone cancer cells, also we see what we were hoping for, which is they kind of peak early and then go back down, meaning that they do try to evade these T cells by clustering up with the other cancer cells. Another paper that we wrote, which was published at PSB this year, had to do with exactly the problem of, if I give you a frame from any of these videos, tell me what knockout I'm looking at. We did this, we put it through GradCam, which is an explainable AI method, and we saw the following, and I'll sort of tell you how we interpreted this after staring at them a long time. So for these safe harbor knockouts, we saw basically that there was a uniform coverage of cells. So all of these sort of red marks here are finding cells that are sort of spread out evenly across the space. So that's what happens in the safe harbor. In the Col5 knockout, we actually found a lot of these cancer cell aggregates, which was kind of cool. In the RASA2 knockout, we actually found a lot of blank spaces, which I thought was super interesting, because unlike Col5, where the T cells are rapidly proliferating, there's not rapid proliferation in the RASA2 knockouts, and so you actually see either clusters of cancer cells and T cells or blank space in these wells. So it ended up doing a really good job. And as you can see, you can imagine going after something like this with a segmentation method. Even if you use the best method in the world, even if I manually annotate this, I'm never going to be able to pull out how many cells there are there unless I have good tracks. Okay, so after doing that work, we went back to the tracking problem. So this is really hard, I think. There's no way to sort of sugarcoat it. Even if you look what's going on in this image, you can see a lot of clusters of cells. You can see cells kind of disappearing and splitting and jumping around. At the end of the day, we figured out, first things first, we need to register, do frame registration on the first step. So we started out by doing that, and then we started staring at it. Also, this is a 3D space. I mentioned it before, but really there's a lot of spaces where these times where these T cells essentially come up beyond the focal point of the IncuSite camera and then go back down, and they look like they're jumping around. They look like you miss a couple of frames with them in it as they move. The cells are complex and heterogeneous. They all look weird and goofy and have funny shapes. Detritus is common. So as you can sort of see, there are some places here where the cells may or may not be actual cells. They may just be garbage, essentially, and they're not moving very much. The shapes change a lot. There's a lot of different morphologies going on. How do we actually accurately identify death or proliferation, which happens a lot? And also, what is the role of fluorescence? How are we going to use fluorescence here? So I'll tell you about our entire all hands on deck thing this summer that we did. So the first thing we wanted to do, which actually we didn't do, it ended up being Julia Carnevale's group, is to develop the data that we needed to be able to benchmark using tracking methods, or these tracking methods. So this was a really cool idea, I thought. It wasn't mine. We basically have a single well, and we use the IncuSite machine to image once per minute. So before we were doing once every 4 minutes, and now we're doing once per minute for 72 hours. So we have a really dense frame rate. And we can subsample that, we can sparsely sample that to be able to get the frame rates that we're looking to see how well we do. Then we can segment and annotate and then manually clean all of these things. And here's actually our manual annotation of a small area of this. And you can see, again, a lot of weird stuff happening, but this is the dense frame rate where things are moving a little slower. And so we do a better job. And I'll say that we did the original segmentation and tracking using a fairly new method called ULTRAC. And I'm going to basically talk about ULTRAC moving forward. Whoops. Let's figure out. Okay. Too many movies. Okay. So we then modified ULTRAC, which, you know, to just say it in very simple English, we forked the GitHub and we added a whole bunch of stuff to the if-else statement. So here are the 4 things we added to the if-else statement, right? We, first of all, sorry, I should say, the way that ULTRAC does this currently is you extend the track for a cell if their masks in subsequent frames overlap. So very, very simple. And we don't have that situation for a lot of cells here. There's a lot of times where the cell masks do not overlap, or there's multiple cell masks overlapping, et cetera. So that's why it doesn't perform perfectly. So the number one thing we added was matching nearby cells that have similar shape and size and are unmatched. Not particularly brilliant, but there it is. The next thing we did was actually build a full neighborhood graph, and then we matched unmatched cells within this neighborhood graph that have similar graphs that are connected to the same cells. This also improved, and I'll say that we're not entirely done benchmarking, but I'll give you some idea of how well these things do. The third thing we did, which I thought was extremely clever, was I have a new postdoc that was like, well, why don't we just use simple diffusion models and make the full video? So she did a full imputation between every single one of these frames, so we can look now at these cells going around very slowly. So full diffusion model between every frame, and that way we can actually pretend we know where and how fast each of these cells are moving. And the fourth thing was actually develop these ideas of latent cells. So we might have, you know, 30, 20 cells here, something like that, but we actually have places in this image where the cells kind of disappear and reappear, and we keep around a whole bunch of annotations that may or may not be that cell showing up again downstream. So to sort of quantify how well we do, originally, when we just use ULTRAC on the 4-minute lag time, we have tracks that are on average approximately 3 frames. And as of EX2, as of our extension to neighbor graphs, so we haven't even done the diffusion between frames, we are already up to tracks that average 14 frames. So we're doing substantially better. It's still pretty bad. We still have about 1,000 frames, and it's kind of shocking that on average, these cells are only tracked for 14 frames. So we're hopeful that the diffusion model will do a lot better. But we also have a way to benchmark now. Okay. So to kind of put it all together, the future work we have involves, well, first of all, imitation learning. What are the local signals that determine the trajectories of these modified T cells? How well do these live cell imaging videos relate to what happens in vivo? This is a huge problem. A couple of months ago, Alex Marzin came to us and said, whoa, we have these amazing T cells, you know, in the live cell imaging. They look great. They're killing all the cancer cells. Everything's awesome. And then, you know, a week later, he was like, oh no, they killed the mice immediately. You know, they're not going to be therapies. And we'd like to understand how that happens. Another thing that I'm really excited to start doing, just like Perturb-seq, can we do this in a screen? So can we actually take a whole bunch of these different perturbations of these T cells, put it in a single well, watch them interact, watch them work together or against each other, and then do post-hoc genotyping to figure out what's going on, and use the tracks that hopefully span the entire video to figure out exactly which genotype knockdown, genotype modification is doing what work. We also want to do active learning. Where can we get excited about T cell behaviors? What do we need to study more? What kind of experiments should we do next? But really the key about all of this, and this was the goal 4 years ago, and it still is actually tell us what the behavior looks like from a basic biology question, and how can we design the right behaviors to create better immunotherapies ultimately. Okay, I think I probably don't have enough time. Okay, well, at some point, I can tell you about sensory neurons, which are super awesome, and we did a whole bunch of stuff to classify movement of mitochondria, but to summarize, we want to engineer T cells with specific behaviors, and we have a lot of other work. Okay, thank you. Have time for some questions? Yeah.
This probably has nothing to do with your talk, but at some point you mentioned it.
There's a mic coming right now. Wasn't sure that was gonna happen. It did.
So at some point you mentioned that these are not killing the cancer cells. So are these like CD8 cells?
Oh,
Not
these
the-
are melanoma, A375.
Yeah, but the T cells are not like typical cytotoxic CD8 T cells, but they're not killing them, but they're somehow causing the cell cycle arrest, I guess.
Oh, I think
How
that,
does it happen? Why do they not kill it and instead stop the cell cycle?
yeah, that's a great question. I think it's because they've been stimulated 6 times. I think they're exhausted. Yeah, that's a good question. Yeah.
Yeah, that was great. It's really always interesting to see. I think a lot of us who work on omics, then we don't make use of a lot of this imaging data. But I guess the thing it makes me think about is like you have you're getting access to this super rich phenotyping and behavior that we have. I mean, maybe there's some signature of that we see in the transcriptome. I don't know, but like, do you have thoughts about like what are the data or methods you would need to kind of bring the two spaces together? It relates a little bit to your selenome.
Yeah.
Bullet point.
Yeah, so I mean, the short answer is there's a ton of sort of multimodal models right now that allow this kind of integration. The short answer is working a little bit with like Christina Curtis and other people at Stanford. The percentage of variance explained, or however you want to think about it, of like the transcriptome versus the morphology of a cell, it's probably max 20%. So I don't know how well we can do. But I mean, we can absolutely build those models. Yeah.
Might be an argument you need both then.
Yeah,
right?
I think you need both. Yeah, exactly. Yeah.
Thanks for the interesting talk. So in one scenario, you mentioned that the T cells do not kill the cancer cells directly; they prevent them from proliferation. And in some scenario, there's one T cell binding to the cancer cell, but the cancer cells still proliferate. So is there a metric or some kind of stats summary on how many T cells there need to be to stop the cancer cells proliferation or to kill the cancer cells directly?
Yeah, so we kind of showed it here. So again, the y-axis here actually represents the change in proliferation log odds for a cancer cell for every additional T cell that's hanging off of it. So we're just assuming it's additive, and we're saying basically if there's one T cell, then your probability of proliferation goes down a little bit. If there's more, it just adds on to that in terms of log odds. Yeah. And similarly with RASA and Col5. So I mean, I think the punchline is, they can proliferate no matter how many cells are hanging off of them, but the probability gets really low. Yeah.
Barbara, I had a question.
Yeah.
You say that some of the cells are disappearing because they're going out of the focal plane. Have you thought about modeling 3D position and this is a projection of 2D?
Yeah, that's a really good question. So that's actually kind of where we started with this. There are a bunch of these really nice imaging setups that allow you to do 3D imaging. It turns out to be much, much, much like 2 orders of magnitude more expensive. So these methods don't really exist in ways that are sort of feasible for us to get our hands on them. I'll say like
That's the data. I mean,
yeah, that's the data.
The model can think something's 3D even if it's 2D, right?
Sure. Or, we could just have a latent variable and watch when these T cells hop out of frame, right? And yeah, impute the rest. Yeah.
Easier.
Yeah.
All right. Are there more questions for Barbara? In that case, let's thank her again.
Hopefully have a presenter.
Yeah, we do have a speaker now. That speaker's name, I'm about to reveal it.
Arashri Gupta.
Yeah, and then we'll get ChatGPT to tell us whatever they can.
Oh, you're online. Okay, perfect. Didn't realize that.
I'm online.
Okay. We will. She's online, so we will hopefully figure this out. So if you share your, let me put back on. It's a dangerous move, wasn't it? Disconnecting.
Are you?
Yes, I can hear you on here. We need to figure out, give us a second to figure out how to get the sound to go to,
Okay,
yeah, no, give us a second to figure out how to get the sound's not going to the speakers currently, so I need to figure that out.
okay, no worries.
I would love to make this full screen, which I think that's going to be as good as we get. Probably
Ah, okay.
right?
Yeah, that's probably gonna do it.
Maybe.
What's your AirPlay code?
I don't know the AirPlay code for this room. Oh, actually, no, you know, I can just do it, but I can just do HDMI on here. Okay, can you try talking again now?
Yeah, can you hear me now?
Yeah, I can, but you're still going through my laptop instead of through the HDMI. I'm gonna see if there's something I can do.
You want to bring a microphone up? Maybe we can try.
Yeah, that's great. That's a good idea.
Okay, Arushi, we're gonna try a little hack here. Okay, try speaking now.
Can you hear me now?
You guys good? Okay. All right. I just gotta get you back on screen. I don't want to touch your laptop because it look complicated. Oh, good. We have Zoom back.
Yeah, same age. Yeah, that's nice. No, I can't, because then the Zoom can't hear her, I think. Because it's the same. I think it's okay. We'll see.
Okay, we got to get her.
Were you getting feedback? No. Yeah. Okay. Wait. Okay, I think you're good to go.
Okay, great. Thank you so much. Hi, everyone. My name is Arushi, and today I'll be presenting my paper on representation learning models for microscopy are confounded by background cells. So within a population of cells, single cells within that population can vary in many ways. I'll give a few examples. They could vary in morphology or the shape of the cell. They could also vary in protein localization, meaning where a specific protein is located within a cell. They could also vary in cell cycle stage, where that cell is within the cell cycle, and so on. Single cell variability is not just noise. It's central to answering many biological questions. For example, let's say we have a population of cells. And let's say for each cell, we can determine the cell cycle stage, where they are in the cell cycle, and we can also determine the localization of a specific protein within that cell. And we notice a pattern that in earlier stages of the cell cycle, the protein will localize to the cytoplasm. And for cells that are in later stages of the cell cycle, the protein will localize to the nucleus. Based on this pattern, we can infer that this protein might be a cell cycle regulated protein, which is a protein that plays an important role in the cell cycle. This is actually really useful to know because dysregulation of the cell cycle is the cause of cancer. And so knowing which proteins are cell cycle regulated and which ones are not can point us towards potential targets for cancer therapy. Now, this is just one example, but in general, single cell variability allows us to uncover regulatory processes without the need for external intervention or perturbation, just by observing cells in their natural state in a population. Given its importance, we want to be able to study single cell variability systematically. And fluorescence microscopy is a key modality for this, because it allows us to visualize the phenotypes of thousands of cells at once at single cell resolution. We can specifically tag
and image cellular components, such as proteins, for a population of cells. And so here's an example of a microscopy scan. And this shows the protein localization of a specific protein, which is imaged here. And we can see that for these different cells, there's phenotypes like cell morphology that's being captured, as well as a protein localization, and it varies from cell to cell. To study single-cell variability in microscopy images, researchers will typically extract features from single cells. Traditionally, researchers would first segment all the single cells in a microscopy scan, and then they would extract previously manually defined features, capturing things like shape, intensity, and so on. And so from this, we can obtain a feature vector for a cell, and we can use this for downstream analysis after obtaining feature vectors for all the cells in a microscopy scan, or for a specific population of cells. More recently, researchers have shifted towards using deep learning methods for feature extraction. The idea is to train a model to automatically extract relevant features from a single cell image. A key detail here is that these deep learning approaches primarily operate on single cell crops, which is a cropped image of a single cell rather than segmented single cells. And so an issue is that these single cell crops contain background information, and this often includes nearby or surrounding cells, as you can see in these examples. This raises a question: are these single cell feature representation models truly learning single cell features? Or are they confounded by background information? This is especially important for the cases in which the phenotype of the center single cell is different than those of the surrounding background cells. But in general, if these representation models are affected by background information, then this affects their accurate use of these models for various downstream tasks and analysis. So in order to test for this, we propose a very simple evaluation framework. And the key idea is that a model's representation for a single cell crop should be the same regardless of the background around the center cell. So regardless of if it changes. So our framework essentially involves doing the following. We first fix the phenotype of the center cell. We then want to systematically swap or mask the background in controlled ways. And if the model features change, then that suggests that they are not robust to background information. In our evaluation, we specifically focus on the phenotype of protein localization. So our specific evaluation setup consists of the following. We select two protein localizations, A and B. And for each localization class, we collect single cell crops such that the center cell localization aligns with either one. We then extract features using a trained model for both sets of the crops. We then train a linear classifier to be able to distinguish between the features of the center cell localization A crops and the features of the center cell localization B crops. And we obtain a classification accuracy. And the idea is to do this across different background settings. So the idea here is that the linear classifier is a probe and is checking whether the model features here contain enough information to distinguish between the center cell localizations A and B. If these features truly represent only the center cell, then the classifier's accuracy should stay consistent no matter how the background changes. So using this setup, we benchmark 3 leading single-cell models for protein localization in yeast single cells. These models are PIFIA, paired cell painting, and DeepBlock. And for each model, we compute classification accuracy across all pairs of 15 center cell localizations. And we report the average of these classification accuracies in our results. So the first thing we were interested in validating with our setup is whether or not this act of swapping backgrounds affects classification performance. We observed that when we swap the background, this can introduce imaging artifacts. As you can see in these examples, the center cell is imposed on these background cells, and they overlap in weird ways. And so we wanted to ensure that this was not interfering with the model features and the classification performance. So in order to test for this, we looked at two different image settings, baseline and same localization swap. In both image settings, we ensured that the center cell localization aligns with the background cell localization. But in baseline, we use natural crops that are unaltered. In single localization swap, we use synthetic crops where we do the background swapping. We ultimately find that same localization swap does not degrade classification accuracy. So across the three models, under same localization swap, the accuracy stays within 3% of the baseline. And in fact, for two of the models, we even see a very slight increase in accuracy. And so this validates that the artifacts from background swapping are not adversely affecting classification performance. So after validating our background swapping procedure, we then asked what happens if the background cells are of a different localization than the center cell? How does that affect model representations and accuracy? So in order to do this, we introduced a third image setting, different localization swap. And in this setting, we use synthetic crops, but we ensure that the center cell localization is different from the background cell localizations. And we find that different localization swap significantly degrades classification accuracy. Specifically, across all three models, accuracy drops by approximately 10 to 15% compared to same localization swap. And so this shows our central claim of our paper, which is that these single cell representation models are very much confounded by background cells. So next we wanted to ask a more subtle question, which is it possible that if the background cells are of the same localization as a center cell, that's actually improving performance? To test this, we introduced a new condition, the mask background setting, where we completely mask out or remove the background around the center single cell. And we're interested in comparing this to the same localization swap setting, where the background cell localization matches the localization of the center cell. We find that background cells do in fact improve classification accuracy when they are of the same localization as the center cell. So specifically across these models, we find that under the mask background setting, accuracy drops by approximately 5 to 7% compared to same localization swap. And so this tells us that these models benefit when the background reinforces the phenotype of the center cell. So now that we've established that these models are definitely confounded by background information, we then sought to examine how does this background reliance actually affect downstream analyses. And so for this, we focused on a task based on the fact that around 15% of yeast proteins are multi-localized, which means that they show single cell variability in their localization. And so a very common analysis or a task to do is for these multi-localized proteins to be able to determine the proportion of cells for each localization. So for example, let's say we have a protein and this on the left is a microscopy scan for that protein localization. And we notice that for some cells, the protein localizes to the nucleus. For other cells, the protein localizes more to the cytoplasm. We essentially want to obtain an estimate like the following on the right, where we can say that 45% of the cells are nucleus localized. For 55% of the cells, we see a cytoplasm localization. And this is really useful for helping us be able to infer the function of these proteins. So again, we want to test the impact of background information in doing this specific downstream task. And so in order to do this, we had the following setup. So for every single multi-localized protein, we extract all of these single cell crops from the microscopy scan. We then also obtain masked versions of these crops, where we completely remove all background information. We then extract features from both sets of crops using the PIFIA model, which is the model in our earlier evaluation that we saw was the most robust to background information across all three. Then we train these protein localization classifiers. So these, for each of the image settings, will predict what the localization of a protein is given the single cell features. And so from this, we can obtain proportion estimates. And so what these consist of is essentially for each of the 15 protein localizations, we have a value that represents the proportion of cells in the microscopy scan that have that localization. So ideally, we would expect that if the background is not relying on if the model is not relying on background information, that the proportion estimates under both image settings should be the same. In order to measure if they're different, we use the KL divergence, which allows us to quantify how much one probability distribution diverges from another. So again, like I already mentioned, we calculate the KL divergence between the original crop setting and the mass crop setting in terms of their proportion estimates. But we need a baseline to compare this against. And so what we end up doing for a baseline is we compare the original crops estimate to a uniform distribution. So in this uniform distribution, we're essentially saying that each protein localization is equally represented for that protein. And so this is obviously nowhere near the case for any of these multi-localized proteins. So it's a pretty high baseline. And so if we notice that the KL divergence between the original and mass versions is greater than this baseline, then that indicates a very substantial difference in the estimated proportions. And we find that this is actually the case for around 15% of multi-localized proteins. So we also did a quick analysis of examining some of the proteins with large KL divergences and seeing whether or not the original crops predictions were more accurate compared to the mass crops predictions. And we found that for 10 out of 10 different proteins, five of them, the mass crops predictions were more accurate. And this is an example of one of those proteins. And so we can see that in the original crop setting, the top two predicted localizations are nucleus and Golgi. And in the mass crop setting, the top two predicted localizations are mitochondria and Golgi. And if we actually look at the microscopy scan on the right, we see that the mass crops predictions are more accurate to the microscopy scan. And so ultimately, we show how this background reliance actually meaningfully impacts the results of a downstream task. And so just to quickly conclude, we propose a framework to test or evaluate single-cell model robustness to background information. And we benchmark 3 different leading representation models for single-cell microscopy in yeast protein localization. And we find that all 3 lack robustness to background information. This has direct implications for yeast single-cell microscopy analysis, especially those that involve single-cell variability, like the one I just went over. And so we really encourage that future work focuses on or takes into account the importance of isolating single cells and not using background information for single cell analyses. Thank you.
Okay, let's see how this works. Questions? Let's see if we get feedback. I'm right here. Fix it. If we don't.
I was curious, all the images I saw were relatively sparse, and have you tried in any models that are probably more reasonably heterogeneous in the density of cells? So, or in cases of tissue models where there's a high level of difficulty of separating out cells, like cells might pile on each other, or like, let's say the colon where it's highly structured, highly dense.
Yeah, that's a really interesting question. So for this paper, we specifically focused on the setting of microscopy images and single cell microscopy, but we do think that our method can more broadly apply to any kind of method in which we have these crops instead of focusing on like a single cell. So in like the settings you're talking about, it'd be interesting to apply this evaluation framework as well, but we just focused on microscopy images for this paper.
Yes, okay, thank you.
So I had a quick question. the jump from nucleus to mitochondrial localization is kind of a big one. Do you have any thoughts about why you get getting these massive differences in the mass setting compared to the unmasked setting?
Yeah, let me go back to that slide. So as in why in the original crop setting, we're predicting nucleus versus in the mass crops setting, we're predicting mitochondria. I think one thing that you can observe, so from this scan alone, like if we're taking crops, so like a nucleus localization is generally just like a small, like we'll see a small like circle of like the fluorescence. And if we take these single cell crops and we have multiple cells that are of a nucleus localization, then we'll see this appear, this like small circle appear like many times in that single cell crop. Versus like in a, so then if it's like a mitochondria or like Golgi localization in reality, in which these localizations have like many of these like circles or like small like fluorescence areas, then it's confusing for the setting where we have, or it's confusing for the original crops model to be able to distinguish between like, are there many cells within nucleus localization, or is it more just like a single cell or like one or two cells with like the mitochondria or Golgi localization? So I do think it is because of the fact that the background information is confusing.
Okay, great. Thank you very much. Can we thank the speaker again?
Feel good, I think. Oh, sorry, did the thing where it stopped.
Presenter mode.
Probably not easily. We can try. So this is Keynote, right?
They're like playing lead. Oh.
Yeah, that's
sure.
yeah, the problem is it's not an extended display, it's
Got it.
right.
Okay, that's fine.
You okay? Yeah.
Yeah.
I guess we don't do plant window actually if we're doing that, because then where was it? It was.
Oh wait, is it shared already? This isn't. No, it's not extended. I see.
Yeah, I think I can't, because otherwise it ends up doing presenter mode on both screens.
Okay, yes, got it. Okay, great.
Oh, we're not. It's not sharing to them. Okay, really all good.
Okay, awesome. Thank you so much. I'm Clara. I'm a senior scientist at Genentech, and today I will be sharing our work on reliable algorithm selection for machine learning guided design. And this was joint work with my colleague, Jiwon Park. Okay, we're gonna start with a guessing game. In this photo, there's an elephant and a human. Who can guess how much the human weighs? It can be ballpark, a range, anything, whatever's most comfortable.
150 pounds.
150 pounds, okay. 75 kilos, all right. How about the elephant? Any guesses for how much the elephant weighs? We can do ranges if we're uncertain.
2,000 pounds.
2,000 pounds, great, thank you. Okay, different question. Who thinks by a show of hands that the elephant is heavier? Yeah, okay, yes, great, good. So we're all pretty confident the elephant is heavier, even though it was harder to form very confident predictions about the individual weights. And so the point here is that we're all using machine learning to make predictions about things, whether it's genes or proteins or cells. But ultimately, prediction is not an end unto itself. We make predictions ultimately to make decisions.
And those decisions can be scientific conclusions, like based on diagnosis predictions, I conclude that the prevalence of this rare disease in the population is 0.1%, something like this. Or the decision can be what cells, what perturbation should I make to a cell to change gene expression? What drugs should I test in the lab to find one with certain properties? So these are downstream decisions that we're really interested in getting to. These are our end goals, and we just think of predictions as a means to that end. So from that perspective, we need more than ever to be thinking rigorously about how prediction error and prediction uncertainty affect our downstream decisions of interest, and beyond just task agnostic kind of notions of prediction error and uncertainty. And so today, I'll be sharing with you how we kind of took this perspective to tackle a problem in machine learning guided design that we call design algorithm selection. So what is design algorithm selection? Machine learning has been used successfully to design novel proteins, small molecules, materials that have desired properties. So this could be novel enzymes with high catalytic activity for a reaction of interest, novel regulatory sequences that modulate gene expression in desired ways, etc. So if you want to use machine learning guided design now, there's a whole zoo of different models, different algorithms, we'll call them design algorithms, that you could be choosing from. So each of these will generate designs from some distribution that it believes has the property that you are trying to get at. And these design algorithms work in different ways. Almost all of them will leverage some kind of predictive model of the property that you care about. So this could be a predictor of fluorescence, gene expression, activity, whatever the thing is you're trying to design for. And this could be a task-specific predictor that's trained on labeled data, or it could be a foundation model or whatever. And then there's almost always some sort of optimization or generative procedure that interacts, usually interactively or usually iteratively with that predictor in order to produce designs that look like they have good predictions. And this could be as simple as like add random mutations, get predictions for the mutants, keep the mutants that have good predictions, rinse and repeat. Or it could be a more kind of modern day generative model, and this interaction could involve actually retraining that generative model to find good predictions more effectively. But this is kind of a loose template that a lot of design algorithms follow. And so the question is, given all these different options, how can you select a design algorithm that will produce successful designs for you? And so this is what we call the design algorithm selection task or problem. And I'll note, of course, that this problem of selecting which algorithm to use for a task has been around since the early days of computer science, and we're probably all very familiar with model selection in particular, where the goal is to fit a particular distribution of data. And philosophically, model selection is in some sense one way to describe the whole process of science. So it's a very long-standing idea that has roots in many, many fields beyond just machine learning. And so I want to emphasize that design algorithm selection is not that. We have quite a different goal. Our goal is not to fit given data, but to generate completely novel data that has a certain property. And so whereas in model selection, as long as you have held out data from the distribution of interest, you can evaluate the fit of any model on that held out data. Here, the whole point of design is to generate novel objects that we've never seen before, we don't have the labels for. And so this is more an issue of forecasting rather than fitting. Before I acquire labels, how can I know beforehand which algorithms are going to produce successful designs for me? Okay, so one thing that you can do is use your favorite machine learning model predictor to get predictions for that property of interest, for that label that you care about. And then what you might do is you might take the design algorithm that looks like its predictions are the best. So for example, that could be these ones with the highest value predictions here. However, this is not a reliable strategy in general because of prediction error. In particular, in design, we're almost always extrapolating beyond the training data. So it's not at all unusual for designs to have predictions that look really good, but when you actually get their labels, they're really low instead of being very high as the predictions would suggest. Okay, so in our work, we present a statistical framework for selecting design algorithms that will be truly successful with high probability guarantees. And the way that we do this is by using a small amount of held out labeled data for example, from training or previous rounds of design, to characterize how prediction error would bias our comparison of different generative models, and then remove that bias appropriately. And so as a teaser plot of what we get is that when a user requests that they want a certain average design label, so that's this x-axis, our method will return design algorithms that have an average design label that's at least that desired value. So mostly above this diagonal y equals x line. In contrast, if you select design algorithms based on just the predictions alone, you'll get average design labels that are kind of far below that y equals x line because of prediction error. Okay, so how do we do this? Just some very quick notation. We're designing objects in the space X. So X will always be like sequences or small molecules or that kind of space. And they have real valued labels Y. So real valued Y is that property you care about, whether it's fluorescence, gene expression, binding affinity, whatever it is. And we have access to some predictor F. So this can be, again, task specific, trained on labeled data. It could be a foundation model, large language model. It could even be the output of some biophysics simulation, something that's nothing to do with machine learning. So this is really just any black box predictor F our method can encompass. We have what we call a menu of algorithms, big lambda. And this could be one particular design algorithm configured with different hyperparameter settings and model architectures. Or it could be a whole bunch of completely different algorithms, or any mix of these things. Each algorithm on the menu generates some distribution of designs, and that distribution of designs induces some distribution over label space called the distribution of design labels, and we call that P Y lambda. And so here I'm visualizing again the predictions, design predictions, which we do have access to, versus the labels, which we do not have access to. We don't know them yet, but of course, this is what actually determines our success. And in particular, we will say that an algorithm, little lambda, is successful if it satisfies a certain success criterion. So this is something that the user gets to specify, and it's that the expected value of some function g of the design labels y, and we call this entity theta lambda, surpasses some threshold tau. So for example, if g is the identity, this just is saying that you want the average design label, which is the y coordinate of these dots, to be at least this value. Or for example, if g is the indicator function, that y is greater than some value gamma, for example, the label of the wild type. This is saying that you want the fraction of designs better than the wild type, which is like the probability mass above this line, to be at least tau. So for example. And I want to note that here we're emphasizing and focusing on the population level behavior of designs. And this is a pretty important conceptual departure from prior work that focuses on prediction uncertainty over individual designs. And so the insight is that in practice, we don't actually need every single design to be a winner. We don't actually even need every design to behave as predicted. We just need some of them too. It doesn't even matter which ones. We need that whole distribution to be right shifted, but we don't care which individuals are responsible for that shift. And so from that perspective, individual or instance level uncertainty quantification might not actually be the most effective strategy for design algorithm selection. Okay, so given the success criterion, our method will essentially construct a subset lambda hat of the menu, such that with probability at least 1 minus alpha for user specified alpha, all selected algorithms will be successful. They will have this, they will satisfy this success criterion. And note, lambda hat may be empty if there are no successful algorithms, or more generally, if our method does not find sufficient evidence that there are any. Oh, okay, and the important assumption is that we do have access to some held out labeled data. So this could be held out from training, or this could be from previous rounds of design. And we'll basically use this to assess how prediction error would kind of distort our comparison of different algorithms. So how do we do this? So the first step is actually to frame design algorithm selection as a multiple hypothesis testing problem. So for each algorithm lambda, we're going to compute a p-value to test the null hypothesis that this population level metric theta lambda is greater than tau or not. Of course, how do we do that? Because we don't have any design labels. What we do have are predictions for all the designs and in that small amount of held out label data. And so from here, we're going to turn to prediction-powered inference. This is a statistical framework for performing valid statistical inference when you have exactly these two ingredients, a small amount of labels, so like real data, in addition to predictions of that label from any black box predictor. Okay, so at a very high level, what prediction-powered inference is going to do is characterize how prediction error would bias a p-value that was computed based on the predictions alone, and then kind of unbias things accordingly. And to see how we do that, let's look at, for example, the average design label. We're going to do something extremely simple. We can rewrite this as the average design prediction, and then this remainder term, which is just the average sign residual between the design label y and the predictions. Okay, so this first term does not have any labels y, so it's super straightforward to estimate. You take a sample mean of design predictions. This second term does have labels, and we do have labeled data, but it's from the training distribution, not the design distribution. So we're going to reweight that data and use an importance-weighted sample mean to estimate that. Okay, so now we have an estimator. That's the sum of two independent sample means. We can just use central limit theorem to derive a pretty straightforward valid p-value for testing if theta lambda surpasses tau or not. And prediction-powered inference can actually handle a broad class of estimators beyond just expectations. So we can actually handle any estimate that is an M estimator. So the minimizer, the population minimizer of some loss that's convex. And things are a little bit more involved here, but the basic intuition is that this minimizer can be equivalently characterized as the average loss gradient being 0. And then we can do the exact same simple trick with this average gradient. We can break it down into a term involving only predictions, and then a term that kind of characterizes prediction error in terms of the bias of these gradients. Okay, so going back to our multiple testing framework, we now have valid p-values for every design algorithm to test if it's going to be successful. And now we can just use any multiple testing correction to select algorithms. And for simplicity, we just went with Bonferroni, but you can easily use any other multiple testing correction that accounts for correlational structure, hierarchical structure between algorithms on a menu, and that will probably improve power. Okay, so I'm going to walk through some experimental results now on simulated protein and RNA design tasks. Recall that our method does use basically importance weights, or these density ratios between the training and the design distribution, and that's an important part of characterizing prediction error. So I'm first going to show results in a setting where we know those density ratios, and then a different setting where we have to estimate them using basically a classifier-based density ratio estimation technique. Okay, here, so in this top subpanel, on the y-axis is the error rate. So this is the rate at which a method selects an algorithm that's not successful. So we want this to be low. And on the x-axis is different values of the desired mean design label, that's the tau. So as you kind of go to the right, the design task becomes more challenging, more ambitious. We want the average design label to be higher. And on this bottom subpanel, on the y-axis is selection rate. So that's the rate at which a method returns anything at all, because if it believes that there is actually no algorithms that are successful, it can just return the empty set. So we want this to be high. We don't want it to be getting the empty set. And so you can see that our method in green is simultaneously very low. Actually, it's controlled under alpha as guaranteed by theory, while maintaining a high selection rate for a broad range of tau. And what that means is that if you select certain tau, you're most of the time going to get design algorithms that actually achieve that or higher. When we move to estimated density ratios, we do lose a theoretical guarantee, of course, but we still achieve an error rate that's much lower than competing other methods, and also a higher selection rate for kind of a broader range of tau. And so that wraps it up. So we formalized this design algorithm selection problem as selecting a design distribution that achieves a population level success criterion. And this focus on population level allows us to not work with kind of individual level uncertainty quantification, but more prediction powered inference way of assessing how prediction error affects population level quantities. And because of that, our method can select successful algorithms with high probability. So a quick thank you to all of you for listening, for my colleagues for giving lots of formative feedback, and thank you to the organizers for organizing this great conference, and open to questions. Thanks.
This was great, and very beautiful slides also. The question I was going to ask about was, so the way you set things up currently, you're selecting some subset of the available algorithms, but I can also imagine settings where it might be interesting to sort of weight them, basically.
Right.
Like I sample which algorithm I'm going to sample from, and then I sample from that algorithm. And I was trying to think through in how you frame things and whether that would ever actually be what you want to do. But I do imagine if you have something about diversity, about wanting, which is maybe a sub question of this, is like thinking about the diversity of the samples as well, which I don't think this framing doesn't cover, right?
Right. Yeah. Great questions. Yeah. So this, what we focus on, we focus basically on a family-wise error rate guarantee. So with high probability, everything that is selected is fine. So if the method returns to you 5 algorithms, any weighting of all 5 of them would be valid. And yeah, it's a good point that people often do care about diversity in X space. So this whole work is kind of focused on Y space, where we don't have labels why we want to be forecasting this. Kind of orthogonally, people also care about X space, which is easier in the sense that you have X, or you don't have to do fancy like statistics and guessing about what X will be. But you can certainly, like, if the method returns to a whole bunch, a non-empty lambda hat, you could pick the one that's most diverse, and that wouldn't change any validity. Yeah, but thanks. Great questions.
Yeah, wonderful talk. Are there any major barriers that prevent this from being used beyond, I guess, the expensiveness of inference, but like as a score for optimizing models?
Do you mean by optimizing models, do you mean improving a design algorithm?
Yeah, models, this and differential.
Oh, that's an interesting question. It's certainly not as framed, I don't think, and then differentiable. But that's a great question as to how to reverse engineer this, essentially, to find the best design algorithm. I think making decisions based on the statistical inference would immediately break everything because of double dipping. Great statistics, but that's certainly a great kind of next line of thought to be thinking about. Yeah, we didn't really address that here.
One more question. Do you have any intuition about like which design method works the best using the approach and why it performs really in this way? Maybe one small question also, how is it related to the performance of the predictor overall?
Yeah, great questions. I'll do the second one first. It's a little bit more straightforward. So this guarantee is always going to hold regardless of your black box predictor. What changes is power. So if you have a really awful predictor that doesn't really allow this method to forecast in a statistically reasonable way what's going to be happening, this lambda hat is probably going to be empty a lot. The method will just be nothing successful, which is not helpful to you, right? But it's not incorrect in some sense. It just doesn't have signal to detect things. Sorry, what was the which design algorithms are best based on this?
It doesn't tell you anything about the why it performs better.
Yeah, not really in a mechanistic or interpretable way. But I think, yeah, that part of the motivation of this is that, like, people will report, oh, this method is best on this giant benchmark. But for any given task, the best thing could be, completely different from like the thing on a leaderboard, right? And so that is kind of the motivation for this, was that we were seeing across a lot of different tasks, the thing that was best is, quite variable, and it's not very clear to tell why.
One more speaker?
Yeah, Joe. She's coming.
Okay. Thought I was gonna have to say someone's name in a very mean way.
We'll see. Oh, right. Lots of that. Okay, so you're on the Zoom now. Is that right? Yes.
So I think which one is it?
What's going to be tough is get on that speak notes. The question is what we got up there. Yeah, I think that's good actually. And the sound is actually the sound is going through mine, so you don't need to unmute. Okay, nice.
I will do my best to keep us on time for a break. So hi everyone, I'm Julia. I'm from the Knowles Group here at NYGC, and today I'm excited to share with you some of the recent work that we've been doing surrounding predicting splicing factor activity from transcriptomic data. So foremost, the key concept that we're focusing on in this project is alternative splicing, which we study a lot in our lab, and which is the mechanism in eukaryotes where one pre-mRNA transcript can give rise to multiple different mRNA isoforms. And this allows for regulatory flexibility for things like the control of gene dosage, as well as tissue-specific expression patterns. Notably, we know that alternative splicing is regulated by splicing factors, which are RNA binding proteins that are known to enhance or suppress the inclusion of exons by binding to cis RNA regulatory regions. And this is of interest to us to study because they can play a key role in disease-related splicing programs. So one disease-relevant example of the influence that splicing factors can have is the STATMIN2 gene, which is essential for motor neuron health in the brain. And typically in healthy individuals, the splicing factor TDP43 will repress the inclusion of a cryptic exon in this gene. But in ALS cases, TDP43 becomes depleted from the nucleus and aggregates in the cytoplasm, and without it being there to regulate it, this cryptic exon is included and results in a truncated protein that leads downstream to motor neuron degeneration. And this is just one example of how splicing factors can act as these sorts of gatekeepers of splicing programs. And so studying their activity is very useful in helping us improve our understanding of the mechanisms of disease pathogenesis. So foremost, it's important to know we have a lot of existing computational tools like Leafcutter, RMATs, MAGIC, and others that allow us to identify splicing differences between conditions of interest. But it's still a challenge to go a step further to actually determine changes in underlying splicing factor activity, especially without perturbation data from the same cellular context. And there have been previous attempts at this problem. So notably DSREG and CatMap are both Bayesian regression approaches that combine differential splicing information with binding protein models to predict splicing factor activity. And both of these approaches perform generally well, but with the caveat that they require limiting their analysis to only splicing factors for which they have good binding models available. So DSREG's performance especially suffers when binding profiles are incomplete or inaccurate. And similarly, CatMap was only applied to simulated data as well as a highly conservative set of 13 splicing factors for which they had very good binding models. In addition, existing methods are all exon-based, and we were interested in trying junction-level features as captured by Leafcutter, with the rationale being that doing this junction-based approach might enhance performance by providing more comprehensive characterizations of alternative splicing events, including things like alternative splice sites, which exon-based approaches miss. So in sum, our aim is to develop a flexible, intron-centric approach to inferring splicing factor activity that is agnostic to binding site information, that generalizes well across cellular contexts, and that makes minimal assumptions in interpreting the underlying biological signal. So there are three core pieces to our splicing factor activity regression inference, or as we're calling it for short, SAFARI workflow. which consist of data preprocessing, multi-class logistic regression modeling, and then SUSY multiple regression modeling for query data. So for most of this, we gathered data for 118 putative splicing factors knockdowns across the K562 and HepG2 cell lines from publicly available datasets through the ENCODE portal. And these knockdown experiments are very valuable in capturing how the loss of a specific RNA binding protein is affecting splicing patterns, which then allows us to infer their regulatory activity. We then followed a fairly standard workflow for conducting differential splicing analysis with leafcutter, where we first extract junctions using rect tools, and then leverage leafcutter, which uses split reads to group introns that share splice sites into intron clusters. And we then perform leafcutter differential splicing analysis for each cell line and splicing factor pair using 2 replicates per condition, which is all we had, which identifies intron clusters with differentially excised introns between the controls and knockdowns, using a Dirichlet multinomial model. And in conjunction with leafcutter, we also perform PCA-based latent batch correction by projecting knockdown samples into the control sample's PCA space, reconstructing the baseline signal using the top principal components, and then subtracting this reconstructed signal to remove both batch and baseline cell state effects. Then, using the leafcutter results and the batch corrected data, we build sparse junction usage matrices, as shown here, where rows are representing splicing factors, columns are representing junctions, and values represent the differential percent spliced in, or psi values, for each pair between the control and knockdown conditions. We can then take these matrices and frame this as a multi-class classification problem by training a multi-class logistic regression model to predict the perturbed splicing factor from junction usage factors in K562, and then validate on HepG2 data, with the model seeing only one example per splicing factor during training before being tested on the second cell line. And we use the hyperparameter training framework Optuna to optimize things like the PCA batch correction strength applied and model hyperparameters such as the regularization type and strength, while optimizing for the multi-class AUROC of this model. And the main reason for ultimately training this model was to try to infer the most significant junctions for making predictions, especially across cell lines, and because we could then use these features to inform predictions for new query data sets. So finally, the key to our approach is in sum of single effects multiple regression modeling, where essentially we set up a multiple regression for each cell line and splicing factor pair, representing each individual experiment sample, where we are modeling the logit knockdown size as being equal to the logit control size, plus the logit difference between the conditions from the other cell line, multiplied by some 1/r vector of coefficients beta, which are the splicing factor activity coefficients we want to learn in this setting. And in this regression, we can then also introduce the learned junction usage coefficients from the best logistic regression classifier as priors. And the way that this works is for each regression, we give SUSY a prior for each splicing factor, corresponding to how much the observed junction usage vector matches the learned junction usage profile for that splicing factor from training. So we're using sum of single effects, or SUSY modeling, which is a Bayesian variable selection method that was designed primarily for genetic fine mapping to allow us to quantify uncertainty around which splicing factors are contributing to the observed splicing changes. And the key intuition here is that typically in genetic fine mapping studies, you will have many highly correlated SNPs, and there's a lot of ambiguity surrounding their individual contributions. But with SUSY, what we can do is calculate credible sets of causal variants, where at least one feature in the credible set is expected to be related to an observed effect within a given confidence threshold. And this becomes valuable, especially when we are dealing with splicing factors, where multiple splicing factors may show correlated activity or cooperative regulation, as SUSY can allow us to identify these credible sets of likely causal factors rather than forcing selection of a single best candidate. And finally, just very quickly, I do want to highlight that once released, Safari is going to be structured as an Xflow workflow to allow users to very easily apply this method to their own query and query baseline data. With the idea being you would just provide paths to your reads or alignments in a sample sheet along with their metadata regarding replicates and conditions, and be able to run the pipeline with very little computational expertise required. So in terms of our key results, first, our best performing multi-class logistic regression classifier achieved an 80.5% ROC AUC score. We ran hundreds of Optuna trials to determine the optimal junction feature set and model hyperparameters for this task. And the best found model was an elastic net model with strong lasso regularization, correctly classifying 95 out of 118 splicing factors when trained on K562 and validating on CAP22. And with this model, misclassified splicing factors were typically spliceosomal proteins from the same or similar families with overlapping junction activity patterns, or those with less junction-based data. So to build on that a little bit, we did some additional profiling comparing the model performances in both cell lines to the non-zero junction counts per splicing factor at the best found intron cluster significance cutoff of 0.1, mentioned in this figure here. And in general, we found that correctly classified splicing factors have 3.4 times more junctions on average, with a subset of 22 from our set never being assigned to a credible set, as shown by the binary representation below the counts here. And we found that in general, correctly classified splicing factors likely had enough distinctive junctions that generalized well across cell lines, that cross-cell line imbalance in terms of the raw junction counts wasn't as significant for their correct prediction. Next, to investigate whether our model is learning biologically meaningful features, we compared the junctions with non-zero coefficients to e-clip binding data for 51 available splicing factors in K562 through the ENCODE portal. And by applying a Fisher's exact test, we found that all 51 splicing factors showed positive associations between the model selected junctions and the experimentally determined binding sites from these experiments, which indicated that the model is prioritizing alternative splicing events directly influenced by the splicing factor binding. As an additional source of validation to confirm the generalizability of this method, we tested our approach on phenol knockdown data released by Rogalska et al. Last year in November, who profiled 305 RPPs using small interfering RNA knockdowns, and fortunately 72 splicing factors overlapped with our dataset from this data. We found that our model generalized well to the HeLa knockdowns, correctly identifying the perturbed splicing factor in the top 10 predictions 84.7% of the time, as the top prediction over 61% of the time. And in general, when visualizing it, the base factor heat map, as shown here, showed a clear diagonal pattern, which is some splicing factors remaining challenging to classify as related to the amount or distinctiveness of the junction level data that they had available. And finally, we were additionally interested in applying our approach to a disease-relevant application data set. And for this, we selected the Answer ALS data set, which is a large-scale database that came out in 2022 that contains induced pluripotent stem cell, or iPSC-derived neuronal data gathered from over 1,200 ALS patients to support efforts to study ALS pathogenesis. And we were interested in looking at the iPSC-derived motor neurons and the bulk RNA-seq from this data set. However, it is worth noting that these samples were cultivated at 32 days, which is quite early based on existing literature exploring limitations of iPSC models. So we suspected that it might be too early to observe significant pathology. We analyzed 409 samples with available transcriptomic data, which consisted of 178 controls and 231 case samples. So here's a clustergram visualization of the splicing factor credible sets that were predicted for each Answer ALS case sample, with the patient site of onset labels shown on the left-hand side. And interestingly, we observed a significant amount of heterogeneity in terms of the predicted splicing factor activity, which is suggestive of different molecular subtypes at play that are going beyond these clinical categories, which have been shown in other studies to be poor predictors of patient pathology and symptom progression. And this kind of heterogeneity is consistent with the emerging view of ALS as kind of a syndrome that converges on motor neuron degeneration through different splicing dysfunctional pathways. So overall, the top most significant perturbations detected included DDX47, SRSF4, and SERENITY2, which had been previously either directly associated with ALS or implicated in pathways contributing to cellular degeneration. We only have time to go into one example, but I do want to focus in on DDX47, or dead box helicase 47, which has been noted to have a protective effect against R loops in cells, which are RNA DNA hybrid molecules that can accumulate at preferred regions throughout the genome, and which act as a source of replication stress and genomic instability. Notably, a hallmark of ALS pathology is the accumulation of cytoplasmic aggregates that contain inert RNA and proteins that are commonly called stress granules. And R loops have been previously suggested to be implicated in a feedback loop with stress granules, where as they accumulate, so do aberrant transcripts, which promotes further stress granule formulation, as shown in the figure here. And the perturbation of dead box hero case 47 could therefore be suggestive of an upstream contributor to ALS pathology that is preceding things like TDP43 aggregation. Finally, I do also want to highlight that we confirmed the topmost differentiated predicted splicing factors in the case samples showed no significant differential gene expression using DEC2, confirming that these predictions are being generated independent of underlying expression levels. So to wrap everything up, we present Safari as a flexible approach for modeling splicing